Okay, everyone. So before we get into some practice questions, I think we could probably take a look at the Operon concept one more time, just because it's not on the AP Daily videos. It is in there a little bit, but I think we could look at the diagram one more time just to understand it in better detail. And then we'll go through some of the practice questions online. So first, let's take a look at the Operon concept. So, whiteboard. Okay. So when we're dealing with the Operon, we remember our acronym PROG, right? P-R-O-G, P-R-O-G for PROG. And with PROG, we have the promoter first. The promoter is it's a region of DNA that codes for, it's an upstream regulatory gene. So that's what we call it. It's a regulatory gene that when you transcribe and translate it, it produces a protein that will help regulate the transcription of the genes downstream. So downstream means down, right? It comes after. So promoter, then is repressor, then is operator, and then is the gene that we're actually focusing on. This whole thing is called the operon. So there's two operons that we need to be familiar with. One is the Lac Operon, and then one is the trip operon. Okay. So let's kind of draw ourselves. So this is DNA right here, and I'm going to just draw in blocks. We have our promoter here. Then we have an operator. Well, we have a repressor, but that repressor actually goes over here. You can think of it kind of like a little piece that connects. Okay. So we have a little repressor. We have an operator, and then we have our gene. Okay. So in the Lac Operon is responsible for breaking down lactose, and this is in bacteria. The Lac Operon helps break down lactose. So how does it do that? Well, first we need to understand that the genes, we should probably remember the order of the genes. It's Z, Y, A, Lac Z, Lac Y, and then Lac A. These three help produce different enzymes that help break down lactose. The way this works is Lac is what we call an inducible, this one's inducible, trip is repressible, so this is inducible operon. What that means is, so once lactose is produced, what happens is, it's actually another form of lactose called allolactose, so let's pretend this is, but we'll just call it lactose. Either way is fine for the sake of, you just need to understand the concept. So if I have the more and more lactose I have floating around, as more and more lactose is produced, that lactose can now stick into the repressor here, and then as soon as it sticks into the repressor changes conformation. Do you guys remember the lock and key model? So then what happens is once the lactose fills in that little gap, with the induced fit model for enzymes, it's kind of that same idea, except it's with a repressor. This repressor protein changes shape, and maybe it starts looking like this instead of with that nice shape. And because it now has the lactose on there, that lactose changes the conformation. Conformation means shape. It's a common word that we use. Conformation is spelled C-O-N-F-O-R-M-A-T-I-O-N. Conformation. The conformation of this repressor changes, and now it can't fit in there. So now what happens is RNA polymerase is now going to be able to transcribe this gene and produce make those enzymes that are responsible for breaking it down. So it's kind of like this is a negative feedback mechanism. Okay, so originally, RNA polymerase isn't going to go down because the repressor is stuck. It's kind of blocking RNA polymerase from going down. And the reason why is because the cell doesn't need to break down lactose because there is no lactose. And as soon as lots of lactose showing up, one of them is going to bind to this repressor over here. And when that happens, the repressor comes off, and then RNA polymerase can go through, transcribe the genes for Lac Z, Lac Y, and Lac A, and then those enzymes can break down lactose. And then once lactose is broken down, this lactose is going to go away, and then the repressor changes shape back to its original shape, and it goes back. And then once again, RNA polymerase can't go anymore. And then that cycle repeats over and over. So it's a negative feedback loop. Okay. So now with the trip operon, it's kind of a similar concept, except this is a repressible operon, and we'll see why. It's kind of like the opposite of the Lac Operon, and the trip operon is responsible for making tryptophan. If you guys remember, tryptophan is one of the amino acids, I believe. Yeah, making tryptophan. So the trip operon helps make trypto like that. Okay. So once again, we have prog. So we have our promoter region. We have our operator. And then we have our repressor protein that binds to it, so PRO. And then we have G. Okay. So in the gene for this, the order of this is E. Actually, I don't remember. I don't think it's that important, but hold on. Let me look through my notebook and just give you the right information for that. Okay. The gene order for the trip operon is it's EDCBA. Okay, it's just in backwards order. EDCBA. I don't think that's as important to be honest, but that's it. Okay. It's a kind of a similar concept. So I know I'm losing space here. So what I'll probably do, okay, here, let me redraw the trip operon a little bit higher up. Okay. Okay, so we have P, oh, that's an R, not a P. P, we have a repressor that's gonna bind here, and it has a little spot for the repressor, or the lactose or whatever. In this case, it's gonna be tryptophan. So we have the operator here, and then we have the gene here. Gene will go, obviously, we just said that the order was EDCBA of the gene itself. So what happens in the tryptophan in the trip operon is that originally what happens is actually in its natural state, the repressor doesn't bind. So in its natural state where there is no tryptophan, then the repressor is actually going to like sit over here. Remember, it's changed conformation. It can't fit in there. So maybe it looks like this right now. And then, of course, we have a little hole over here. But what happens is as as RNA polymerase goes through and makes, the proteins, makes mRNA for these genes over here, and then that gets translated into the proteins that make tryptophan. Well, once that happens, that tryptophan, these little tryptophans appear, and then those little tryptophans can come over here to the repressor, bind to the repressor, and well, now what happens? The repressor changes conformation back to its original square conformation, and then that shape can bind here. So when that happens, so really this diagram should look like this, okay? So now, obviously, RNA polymerase can't go through anymore. It's blocked. So once again, it's a negative feedback loop. The thing you have to remember is that with the Lac Operon, when lactose isn't present, so if lactose is its natural state is okay, so it'll look like this, right? In the Lac Operon. So this is the Lac Operon. When the repressor is bound to the operator, that means that there is no lactose. Okay, so that's kind of its natural state. In the trip operon, when its kind of natural state is when, once again, there is no tryptophan bound to the repressor, and the repressor is alone. So that means the operator and the repressor are kind of separate. Okay, so once tryptophan binds to the repressor in the trip operon, then it can change conformation of that. But you just have to, they're kind of opposites of each other. So this is a repressible versus Lac is inducible, and the names are exactly what they suggest. But what the important thing is also that they're both negative feedback loops. Hopefully that clarifies the Operon concept a little bit better. And it's, I think for this year's AP exam, it's more kind of understanding how the concept behind it and being able to apply it rather than, brute memorization. I don't know how much that would help. I think it's more important to kind of understand the mechanism by which it works. Okay, I'm going to stop sharing this whiteboard over here. Now I'm going to go ahead and share the screen and go through some practice questions. Okay, so some of these we don't need to know, but we can try answering them. The main goal of the Human Genome Project Well, the Human Genome Project kind of it sequenced the entire human genome, right? It went through all the DNA and it figured out what was responsible for each, what section of DNAs were responsible for what genes and that kind of thing. So the answer should be this one: determine the entire sequence of human DNA. Okay. The specialized field of studying whole genomes, including genes and their functions, is called probably genomics, because that's, remember, genome is in the word, the genomics, okay? Which best describes the impact that the Human Genome Project has on society. Well, if we're able to figure out what the entire human genome looks like, then we can figure out, okay, this gene is responsible for this one's responsible for this. So with that, this is probably the best answer, because it helps scientists determine that certain genes can cause diseases. Good, so that's the right answer. So what is a genome? We already said it's a complete set of genetic material, right? Yep. And remember, each cell, this is another important in AP biology, is that each cell has the entire human genome, okay? So your skin cell has the DNA for like your entire DNA, not just for that skin cell. It's that the reason why it's a skin cell versus something else is because remember, there's regulatory mechanisms and signal transduction systems and all that kind of stuff, transcription factors, everything that helps in differentiating that cell into a skin cell. You know, my eyes have the amount of DNA in my eyes is the same amount of DNA in my skin. Okay, it's the exact same, because each cell has the entire genome. Okay, which of the following is a true statement about natural selection? Well, individuals can evolve. Remember, we talked about that. It's populations that evolve over time with response, and it's generally a slow process, gradualism, right? Acquired characteristics, I'm not sure what that means. Well, variation does exist, but I think this is probably the right answer. Variation exists in a population, remember, because variation is kind of the foundation for natural selection. If you don't have variation, you know, nature can't select on which individuals are more fit. So what happens is sometimes, you know, individuals in a population are more fit because they have variations that help them survive and reproduce, and then they can pass those genes on to the later generations. So it's not that the individuals are evolving. I want to clarify that, but really variation, genetic variation specifically, is the foundation for natural selection. So that's right. Okay. A bird that can easily outcompete other birds for food and that can produce many eggs has high. So remember, this is fitness. Fitness we just talked about that, right? Fitness is ability to survive and then reproduce, because you want to be able to pass your genes on to the next generation. That's fitness. Yep. Okay. Genetic variation can arise from a random change in the DNA of a gene. This change is called a, well, the picture kind of gives you the answer, but it's mutation, right? Yeah. Remember, there's there's two main types of mutations. One is a substitution, and then one is a frame shift mutation. So a substitution or point mutation is where, remember, you just have one nitrogenous base that changes. And then if that happens, it's generally not that big a deal, just because, you know, everything else is correctly done. But an important example of this is sickle cell anemia, and obviously that's not a very small, that's a pretty big consequence. So generally speaking, substitutions affect only one amino acid. The rest of the reading frame isn't affected, so everything else is generally fine. And then the other is frame shift. So frame shift mutations occur from either an insertion, where you insert an extra nitrogenous base, or deletion, where you delete an. So then the problem is, remember, so once we have DNA, let's say our DNA is A, T, C, 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 G. Remember, it's read in threes. Each three makes up an amino acid. But if this is DNA, to transcribe it into RNA, we have to make this what? G, A, G. Sorry, I did this completely wrong. That's a T. That's a T, A, G, 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 C. So, oh, okay, I'm sorry. I don't know what I'm doing. I was doing DNA replication. We're doing RNA. We're going to RNA. So we need to make this a U, A, G, 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 C. Now this isn't realistic because we know that UAG is one of our stop codons. So once this happens, Obviously, once DNA is transcribed into RNA, it's translated into protein. Now, I don't have the codon chart in front of me, but this would become a stop codon, so that's not really productive. But let's pretend this wasn't a stop codon. Let's say that this was something, and then GGC is something else. So let's just say it's arginine for, it's not arginine. It might be, I don't know. I don't have this chart. But let's just pretend this is arginine, and let's just pretend this is methionine, okay? Now, with the substitution mutation, you know, if I change this to a T, right, then this becomes an A, and we don't, it's just one amino acid that we change. Everything else is fine. But then the problem with an insertion or deletion is if I were to insert, say, a G here, then I'm inserting a C here. And now what happens, the entire reading frame is shifted. So UAG becomes methionine, CGG becomes something else now. Maybe it's, I don't know, leucine, okay? And then now the problem is everything else after that is shifted. So you're gonna have something that's it's generally much worse to have a insertion or deletion. And obviously deletion, you could argue the same thing. Okay, so another thing is that might happen is as a result of insertion or deletion, you might reach the stop codon earlier than expected. And if that happens, then obviously, oh, I stopped sharing screen. And if that happens, then you're gonna, you know, your amino acid isn't gonna be coded anymore. You're gonna stop producing, not amino acid, your polypeptide is gonna stop being produced right then and there. Anything else I can think of relating to that? I think, you know, once again, if you just kind of understand the concept and work through it, that's probably a better way to approach it. Okay. A condition in which there are barriers to successful interbreeding between individuals of different species in the same community. So remember this, barriers to successful interbreeding between individuals of different species. Okay, so different species cannot reproduce with each other. So this is reproductive isolation. That's just a definition we need to know. It's reproductive isolation, and that's what leads to speciation, right? Because if population A can't breed with population B, then they're reproductively isolated, and that means over time they'll develop into two separate species because they can't breed and produce fertile offspring. That's the definition of species, to be able to breed and produce fertile offspring. Okay. Maybe you were tempted by geographic isolation, but there's nothing about physical, you know, barriers separating the two communities. And also, yeah. And just another note, geographic isolation is like when you have a geographic barrier, that's allopatric speciation, I believe. Let me confirm that with my notes real quick. I believe that that's allopatric speciation because, well, that's just the definition. Yeah, allopatric speciation is evolution of new species due to geographic isolation. So once again, geographic isolation occurs because of a lack of gene flow. And remember, if we're doing Hardy-Weinberg, we do want some sort of geographic barrier because we don't want gene flow in Hardy-Weinberg equilibrium. We don't want, you know, migration between populations and this gene to influence our allele frequencies for another. That's in Hardy-Weinberg, and we'll get to that probably. Okay. The difference in sparrow songs among sympatric species of sparrows are an example of, well, sparrows that aren't geographic, they're not, I don't know what convergent has to do with this, but mechanical isolation has to do with, you know, the actual mechanics of, you know, the two individuals having intercourse. And clearly here, we're talking about singing, so I don't think singing has to do with the actual mechanics of intercourse. So, so that has to do with like your genitalia, and this, you know, sometimes genitalia are too big or too small and they don't fit. So this is behavioral isolation because it has to do with the behavior of the sparrows. Yep. Okay. Two allopatric species of sparrows have similar niches when compared to two sympatric species of sparrows. What could be a good explanation for this? Well, maybe, let's see. The allopatric species have no competition with one another, so therefore can occupy similar niches. That doesn't really make sense because if you have the same niche, that means you have the same rule. So that means if we're both, if me and some other individual is both fighting for for dinner in the middle of the table, then we both have the same niche, and obviously we're in competition, so that would make sense. The sympatric species are usually mechanically isolated from one another. Maybe, we don't know. The sympatric species diverge from a common ancestor, potentially. The allopatric species are behaviorally isolated. Okay, well, here's what we do know. These allopatric species of sparrows, oh wait, I'm sorry, maybe it is A. I misunderstood the question. Okay, so just to clarify, allopatric species of sparrows are separated by the barrier, right? So we have sparrows, sparrow A and sparrow B over here. But then there's very similar niches if you compare the sympatric species of arrows, sparrows C and D. These two are able to have similar niches, and they're because they don't have competition, because there's a barrier here, right? So it's like two different environments. Whereas species C and D have probably much more different niches because it's not ecosystem, it's not like productive to be competing over the same things. So it's probably A is the answer, because the allopatric species have no competition with one another because they're in different environments. So I'm thinking the answer would be A then. Yeah, okay. Which of the following is not involved in natural selection? Okay, so this would be a good time to talk about Hardy-Weinberg as well, because there's, let's answer the question first. Okay, which of the following is not involved in natural selection? Well, obviously, we talked about variation has to be involved in natural selection. Differences in reproductive success has to, because otherwise nature can't select on the more fit or the better, but it's not necessarily better, but more fit individuals is the right terminology. And obviously there has to be competition, otherwise there's no meaning for fitness. What does it mean to be more fit unless there's competition? So obviously breeder selecting traits, that would be artificial selection, because a breeder is doing that, not nature, so it would be D. So now I think it would be good to talk about the few mechanisms of evolution. And I know we haven't gotten to a Hardy-Weinberg question yet, but I don't know if there will be. So I'm just going to take this opportunity to do that. So kind of the way I think of it is it's kind of a two-sided approach. So actually, I'm going to draw this out. So let's draw a little table here. So we know that condition, so this is conditions for Hardy-Weinberg, HW, and then this, and on this side, I'm going to write the mechanism of evolution. So for each mechanism of evolution, you want that opposite for Hardy-Weinberg, because in Hardy-Weinberg equilibrium, you don't want that population to be evolving. So remember, we have five conditions, so we have 5 mechanisms of evolution. So one, clearly in this question we see is natural selection. So natural selection is a clear mechanism of evolution. So obviously in Hardy-Weinberg, we want absence of selection, right? We want absence of selection, and absence of selection essentially means there's no benefit in reproductive success, and there's no competition necessarily. There's no so in these two things. So there's no reproductive success just because this person, yeah, 2 individuals aren't necessarily one isn't necessarily more fit than the other and more likely to survive and reproduce. That's the idea. So there's no natural selection. So mechanism of evolution is natural selection. Hardy-Weinberg condition is lack of that. Okay. The second one, another example we saw already today is mutation, right? If you have a mutation, that's going to lead to variation. So so mutation is a mechanism of evolution. So the condition for Hardy-Weinberg is no net mutation. So no net change in DNA. Okay. So remember, everything that's on this column is the opposite of what's on this column. Okay, so these are opposites. Now, the third, we talked about gene flow, right? Migration. Gene flow will result in evolution because you're introducing new genes into the new alleles and new genes into the gene pool. It's probably more likely alleles you're introducing rather than genes. Okay, so the reason for gene flow is migration. So the condition for Hardy-Weinberg would be no migration, because if you have migration, then you have gene flow. Okay. There's 2 more we haven't gotten to yet. And then So similar to natural selection, there's sexual selection. Sexual selection happens because there's non-random mating. So sexual selection is the same thing as saying non-random mating. So that implies that in Hardy-Weinberg, we should have random mating, because otherwise, if the girl likes boy A better than boy B, then the girl's going to select boy A. So we don't want that. We want it to be an equal chance for boy A and boy B when we're looking at equilibrium. Obviously, in the real world, that's not how it works. So there's sexual selection. Okay. And then finally is the last mechanism of evolution is genetic drift. And genetic drift happens when you have small populations, right? So on Hardy-Weinberg, we want a large population. And just to review, genetic drift is when in a small population, you could have something like the bottleneck effect or the founder effect, where there's all of a sudden you have genetic drift has to do with, like, random. It has to do with a random change in, like, the frequency of a particular allele. Just because the population is so small, you don't have these other factors that account that can kind of temper it out. Because it's such a small population, you have increased death rates, and it's less genetically diverse generally. So that will lead to problems when you're dealing with Hardy-Weinberg. So that's why we have large population. Okay. So now let's take a look at the next question. So hopefully that clears up both Hardy-Weinberg and evolution a little bit. I think this is a nice way to connect the two. Okay. Which of the following is an example of gene flow? Perfect. So we said that gene flow is dealing with migration from one area to another. So let's see. Subordinate wolf successfully challenges the alpha male and mates with pack females. That looks like sexual selection more than gene flow. The young stallion is driven out of the herd by alpha stallion and begins its own herd in another location. That's actually the founder effect. So I would say that's actually genetic drift, probably. Probably not gene flow. I think this one makes more sense. A deer picks up spores from a plant as it grazes and deposits them. That's the migration of these spores from location A to B. So there's gene flow going on. This one here, sure, it is a new location, but the problem with it is there's not an existing population. He's starting a new herd. So that's actually the founder effect leads to genetic drift, right? Because you have a small population. So you want to, I think the answer would be B. Oh wait, so it's not. It's this one. I think that this might be wrong, personally speaking. Let me search this one up online to see if there's a different answer, because I think the logic that I just provided is pretty solid. This seems like it's founder effect. I think. Oh wait, I'm sorry. Maybe this might be the better answer. Just because here it's saying that it's depositing them to the next plant, so next to that plant. So obviously that's not gene flow from one location all the way to another. Okay, so I would say that this is both gene flow and genetic drift. The young stallion is driven out of its herd and begins its new herd. So it's gene flow because you have alleles migrating, but also genetic drift because you have the founder effect, right? So that's that. Okay, I think that is the right answer. Moving on. James Hutton's, okay, we don't know that it's James Hutton, but we know about the theory of gradualism and then uniformitarianism. I'm not sure exactly what that is, but I think uniformitarianism, just looking at the word, it has to do with populations being uniform or something like similar. I don't know. Let's read the question and see if we can figure it out. Played significant roles in the development of evolutionary theory because together they influenced Darwin by persuading him that, well, Darwin's writing was about descent with modification and evolution with natural selection, that kind of thing. So I think, let's see, occurrence of continental drift provides an explanation for the discovery of similar species on different land masses. That doesn't really have to do with gradualism so much, right? And it kind of explains evolution, but it's not the main thing, I think. Earth is old enough that changes through natural selection could account for evolution of species. Potentially that's an answer choice. I don't know if that's the best one. The common ancestry of all living organisms explains their fundamental similarity. That's a very weird answer choice. What is the fundamental similarity? Let's I don't know if that's the best answer choice. And transitional forms in the fossil record provide links between distinct species. Probably this is the best answer choice, but it could be this one over here as well. I think it's this one though, because it talks about gradualism with, like, because you have fossil evidence with geological evidence with transitional forms over time, so that's gradualism. Uniformitarianism, probably, let's search that up. No need to guess. Let's just search it up. Uniformitarianism has to do with changes in the Earth's crust during geological history have resulted from the action of continuous and uniform processes. Okay, well, that would be transitional forms in the fossil record because it's a continuous form rather than uniformitarianism. So gradualism and uniformitarianism are very similar, and they contrast to the other two, which is catastrophism, which we didn't learn about, but it's very similar to punctuated equilibrium, I think is what we learned, right? Yeah, punctuated equilibrium, where you have rapid evolution after long periods of stasis. So generally, I think gradualism and uniformitarianism work better, but that's from kind of the evidence I've seen. But I think this one is the right answer because this deals with gradual transitions and fossil records. So apparently this is the right answer. Earth is old enough that changes through natural selection could account for the evolution of species. That does make sense if you look at uniformitarianism, that Earth is old enough that it could account. I don't know if this is the best question. I think this one makes sense, but once again, we haven't studied uniformitarianism. Maybe it's this part that makes this answer choice wrong, the links between distinct species. Maybe that's what makes this wrong. Maybe it's the fact that you should probably be looking at kind of the same population over time rather than different species. Okay, well, this is the right answer. It makes sense, I guess, with the idea of uniformitarianism. Okay. Species that are not closely related and that do not share many anatomical similarities can still be placed together on the same phylogenetic tree by comparing their plasmids. I'm not sure why I would compare their plasmids, because not all organisms, it's bacteria, have plasmids, right? Mitochondrial genomes, maybe that could make sense. Homologous genes that are highly conserved, and then homologous genes that are poorly conserved. Okay. I think I'm not exactly sure. I think it's probably, I just had to take a guess. Homologous, I feel like it's probably one of these two answer choices, but I'm not sure whether it has to do with poorly conserved or highly conserved. Let's think about it. If homologous genes are highly conserved, then we can group them together. So probably if they're poorly conserved, then we probably wouldn't be able to compare them. So I'm thinking it's this one, but I don't know what the answer is. Okay, so this is the right answer. It's homologous genes that are highly conserved. And because they're highly conserved, we can kind of group them together. That's the rationale behind this, I think. So over evolutionary time, many cave-dwelling organisms have lost their eyes, tapeworms lost their digestive systems, whales lost their hind limbs. The natural selection, how can it account for these losses? Let's see. Maybe it's this one. Under particular circumstances that persisted for long periods, each of these structures presented greater costs than benefits. So obviously, natural selection will say, well, if it's making the organism less fit, then that organism is less likely to survive and reproduce. So maybe those digestive systems and hind limbs were a greater cost than benefit. And that's the right answer. Okay. If Darwin had been aware of genes and of their typical mode of transmission to subsequent generations, with which statement would he most likely have been in agreement? Okay. So it should probably combine something with his theory of natural selection and then combine that with this idea of genes, right? If a natural selection can change one's gene frequency in a population over the course of generations, and given enough time and enough genes, Natural selection can cause sufficient genetic change to produce new species from old ones. Maybe. Let's just look at the other ones. If an individual's somatic cell genes change during its lifetime, making it more fit, then it will be able to pass those genes on to offspring. Maybe. Let's see. An individual, but okay, this one is probably wrong because it's the somatic cell genes, because those are your body cells, right? If one of those change, you're probably not passing that on. It has to probably be one of your gametes, right, that are being mutated rather than the somatic cells. If an individual acquires new genes by engulfing or being infected by another organism, then the new genetic species, no, that doesn't make any sense. A single mutation in a single gene in a single gamete will, if perpetuated, produce a new species within just 2 generations. There's no evidence to support that. I think the best answer choice is this one, but let's just read through it one more time. If natural selection can change one's gene frequency in the population over the course of generations, okay, then given enough time and enough genes, natural selection can cause sufficient genetic change to produce new species from old ones. So through maybe reproductive isolation for whatever reason. So I'm thinking probably this is the best answer. Yeah. So, you know, over time, if there's enough mutations, then that will result in reproductive isolation and speciation. Okay. Natural selection is best based on all of the following except, well, I think the best, so we talked about how this is probably true. The best adapted individuals tend to leave the most offspring, so that's fitness. Individuals who survive longer tend to have more offspring, probably, because if you're living longer, you have more time to make babies. Populations tend to produce more individuals than the environment can support. Maybe. Yeah, that seems to be true. I think this is the wrong answer. Individuals adapt to their environments and thereby evolve. It's not individuals who evolve, it's populations who evolve. So it's probably this one that's wrong. Yeah. Remember, populations evolve, not individuals. That's a very important concept. It's a recurring theme, I think. During a study session about evolution, one of your fellow students remarks, giraffe stretched its neck while reaching for higher leaves. Its offspring inherited longer necks. As a result, which of the following will correct the student's misconception? Well, I think it goes back to our thing about individuals, right? Individuals don't evolve, it's populations who evolve. So characteristics acquired during an organism's life are generally not passed on through genes. That makes sense. Because remember, we talked about how the mutation has to be in the gametes, and then that can be propagated down. But once again, natural selection is more about the more fit individuals who are more adapted to their environment will be able to survive and reproduce more and have more babies and pass on their genes. Yep, so that's the right answer. Okay, next one. Morphological, actually, let me take a look at how many questions there are. Oh, wow, there's 63, okay. I mean, I'm here, so I'm just gonna record this and you can go through them as you wish. Morphological and genetic comparisons group 30 species of snapping shrimps into 15 pairs of closely related species. Okay, so we have morphological and genetic comparisons, and they're grouping 30 species of shrimps into 15 pairs, okay? In Allocatric Speciation, a new species may arise in the same area as an existing species, where in Allopatric, we talked about, you know, how there's a geographic barrier. What is the best explanation for the fact that one member of each pair lives on the Atlantic side of the Isthmus of Panama, whereas the other member of each pair lives on the Pacific side? Well, here's the thing. We talked about how, you know, if you're in the same location, if you're on the same side, then there's competition. You're filling the same niche. That's not productive. So it makes more sense that the same species, in like similar species, one is going to be on one side and one is going to be on the other side, because now they're, you know, filling the niche for that particular environment rather than having to compete. Let's see. Different predator pressures in the Atlantic and Pacific selected differences resulted in the reproductive different predator pressures in the Atlantic and Pacific selective differences resulting in the reproductive isolation of these species. That could make sense, I guess. The pairs of species arose by allopatric speciation when the isthmus of Panama separated their ancestral species. That could make sense because remember, allopatric speciation is that barrier that separates the two species. The 30 species of snapping shrimp evolved by sympatric speciation before the Panama formed. That's unlikely. And then 15 pairs of species illustrate the pattern of, I think it's this one, because remember, this geographic barrier is resulting to speciation through reproductive isolation. Yeah. Okay. Mice that are homozygous for lethal recessive allele die shortly after birth. Okay. In a large breeding colony of mice, you find a surprising 5% of all newborns die from this trait. Okay. So the lab records show that the same proportion of offspring have been dying from this trait in this colony for the past 3 years. So how much you explain the persistence of this lethal allele? So generally we would say, well, nature would select for the fit individuals and less and less would die over time, right? But it seems like it's the same number who are dying. Why is that happening? So if it's homozygous recessive, it can't have a reproductive advantage. That doesn't make sense. Obviously, they're dying. Yeah, they're dying, so that doesn't really make sense. What about this? This might make sense. There's some sort of heterozygote advantage and perhaps selection against the homozygous dominant. So, well, if there's heterozygote advantage, that keeps the little R allele, the recessive allele, alive, kind of. So it can continue being passed down. So if there's a heterozygote advantage, then that would support natural selection, and that could account for why that lethal allele is still there. Obviously, homozygous recessive is bad, but heterozygous advantage could make sense. The rapid reproductive rate is coupled with a large mutation rate, which results in multiple copies of this lethal allele. Doesn't really make sense because the mutation, the ones who are homozygous recessive are dying, so it doesn't really make sense. So they can't reproduce. Because this is a diploid species, the recessive allele cannot be selected against when it is in the heterozygote. That doesn't make sense. It can be. So it's this one, I believe, is the answer choice. Yep. So one general pattern that will maintain two related groups as separate species requires their gametes to not combine. Okay? These are called prezygotic barriers. We learned about that, right? Select the one example here that does not describe this type of isolating barrier. So we have to remember which ones, like this is kind of by definition as well. So temporal isolation is clearly prezygotic because if two species are mating at different times in the year, they can't even form a zygote. Behavioral isolation is also prezygotic because, once again, it has to do with mating. So if it's after intercourse, then it's supposed zygotic. If it's before intercourse, it's prezygotic. So these three are all before intercourse, before they're able to even have sex. This hybrid breakdown is after sex. It's after intercourse. So the breakdown of hybrid inviability, hybrid breakdown, those are all post-zygotic. So that's why this is the answer. Yeah. In particular, bird species, individuals with average size wings survive the storms more successfully than other birds with longer or shorter wings. So this is, we didn't learn about this, but you can kind of think of this as like a normal distribution. If you have a normal distribution and then there's a selective pressure, and then after a storm, it only increases it, then that would be stabilizing selection. In the AP exam, they're probably going to ask you like more of a graphical question and let you apply that knowledge and kind of figure out, okay, what's happening rather than the actual definition. The definition is not that important. What's more important is you understand that, okay, well, if I have a pretty centered population, a pretty evenly distributed population, and I have all of a sudden now I have more average size than rest, then this is probably when, so then what happens is that the pressures actually, if you were to draw the pressures, the selective pressures are pointing downwards here. Disruptive directional selection would look like this. So all of a sudden after the storm, if you now have more longer sized beaks, then it would look like this. There's a directional shift. And that's because there's a selective pressure on the left side. You could also argue the other way, and that's perfectly fine. That's also directional. And then the other one is a disruptive. So disruptive happens when you're selected. The average value is selected against. So if you have the average value is not selected for, then what happens is this becomes, starts looking like this. So now you have two kind of now two new average ones, because two modes, I guess you could say in this case, because the average was selected against. So this is disruptional selection. I think those are the three big ones that would help to know. Once more again, it's more about the concept than memorizing it. So yeah, stabilizing. Okay. Which of the following statements describes the concept of punctuated equilibrium? So we talked about that, right? So we have you have like sudden evolution, and then you have lots of stasis. Sudden evolution, lots of stasis. So this is the correct answer choice. That's just a definition question. If we were to ask ourselves, what is this one? This is gradualism, right? It's gradualism because it's happening over a slow, continuous process, and also uniformitarianism, or whatever that was that we saw in the previous question. So it's the answer to this one. Punctuated equilibrium is this answer choice here. Yep. Okay. The condition in which there are barriers to reproduction between individuals of the same species because they're separated by a portion of a mountain. As soon as we see mountains or rivers, generally that's geographic isolation, because there's a geographic barrier. So the answer is geographic. Yep. Okay. Which of the following statements is correct based on the info shown in the cladogram below? So we have an outgroup here. The landslide is the outgroup. Okay. It seems like the turtle and the leopard are very closely related, then the frog, then the base, then the lamprey. So let's see. The turtle and frog are more similar to each other than the turtle and the base. That would make sense, right? Because the turtle and the frog share a more recent common ancestor. That's their common ancestor is more recent than the common ancestor shared by turtle and base. So this could make sense. Okay, I'm going to eliminate this answer choice. The base is extinct because we don't know that. Frogs have existed for a longer time. Once again, we don't know that. We don't know whether frogs have existed for a longer time than bats necessarily. This is more about kind of evolutionary relationships. Generally speaking, the further up you go, that's generally chronological, but not always. It's more about the evolutionary relationships with kind of their common ancestors. The frog is more closely related to the bats than to the leopard. That's not true, because the frog, actually, that could be true, but I think the better answer choice is this one. The turtle and the frog are more close than the turtle and the base. But let's check the answer. Yeah, because that's the right answer. Because the turtle is closer to the frog than to the base. I know that you could probably, with that argument, you could say that this is true, but the problem is, generally speaking, once you have that common ancestor, I would look above that. Okay, the frog is closely related to the turtle and the leopard, more than it is to the base, because there's this derived characteristic that sets it apart from the other ones. So that's why the answer is this one rather than this one. So you want to make sure you think about those derived characters. Horse and donkey mate and make a mule. Are mules, so mules are still, they can't reproduce. Are they species? Well, no, because remember, a species is members of a population that can interbreed and then produce fertile offspring. So since they can't reproduce successfully, they're not a species. So this would be the right answer, I believe. Yeah. Okay. Which of the following is the least inclusive? Okay, so we haven't fully covered this yet, but the acronym is this is the one I use. Do kangaroos prefer cake or frosting, generally speaking? Do kangaroos prefer cake or frosting, generally speaking? So we have domain. I'm not going to write them out because that'll take too long. Domain, kingdom, phylum, class, order, family, genus, species. Okay. Least inclusive. Most inclusive would be domain. This is least inclusive. So species is least inclusive, because it's, species is just one species compared to the entire genus or anything else. The species is the right answer. Okay. Randomly selected group from a family. would show more genetic variation than a randomly selected group from a family. Okay, so something from a group selected from a family would be more varied than a group selected from anything else below family, because anything else below family is more specific. It's more, it's less inclusive. So it would have less variation. The higher up you go, the more variation. Think about all the variation in the eukaryote domain, right? So that's why this would probably be genus, because genus is below family. And we probably don't need to know this for this test, but I think it's, you know, nice to just go through these questions. Okay, using the cladogram, a common ancestor of amphibians and ray fin fish and amphibians. So if you look down, the place that they meet is the ray fin fish and amphibians. Their common ancestor is at this node right here. Their common ancestor had bony skeletons and had vertebrae, but not 4 limbs. So they would have a bony skeleton, it's true, but they wouldn't have anything above that. So it makes sense that the answer would be this one right here. So you, like, we want to, you know, draw the lines, connect the nodes, figure out what the node of the amphibians and the ray fin fish are. And that's right. Good. Okay, moving on. The wing of a bat and a human's arm have different functions and appear very different, but the underlying anatomy is the same. And so these are homologous structures, not analogous structures. So remember, you want to associate homologous or homologs or homology, same thing. Homologous structures have to do with divergent evolution. When you have, you know, when you have a common ancestor, like when it comes to evolution, like genetic relationship, they have a common ancestor, but they diverge because they're now in different environments or different circumstances. The other one is analogous structures. Analogous structures happen due to convergent evolution, when you have, you know, they have different ancestors, but they have similar structures because they came together to the same environment. So analogous is convergent, homologous is divergent. This would be homology because they have the same anatomy, so that means they have like the same ancestry probably, but their functions are different. So that's why they diverged, because the functions are different. It's homology, yeah. Okay, in natural selection, the selective agent is the environment, right? The environment is selecting. It's not, humans and breeders would be artificial selection. Mutations aren't the ones selecting. Mutations are kind of a mechanism, right? We talked about the 5 mechanisms of evolution. Mutations are a mechanism of evolution, and generally through natural selection. So environment is the selective agent. A measure of how commonly a particular allele occurs in a population. Well, this is Hardy-Weinberg. It's just the definition. It's allele frequency. It's the frequency, how commonly it occurs in the population, and obviously it's dealing with allele, so it's allele frequency. The more genetic variation a population has, the more likely the same individuals will. So if there's more genetic variation, generally that's better for the population, because you can survive diseases, right? Because, you know, if there's a disease that kills off, that affects only a certain number of people with a certain number of mutation, then the other ones with the other mutation would survive. Whereas if you were, you know, it's like a homogeneous group, then that's problematic, because that would wipe out the entire population. So if you have more genetic variation of population, it's more likely in the same individuals that they will, I think survive is the best answer, because individuals don't evolve. And like we just said, it has to do with like the disease idea. So I think, yeah, survive is the right answer. Natural selection cannot occur without what? Well, unlimited resources doesn't make sense. So this can't be that. So natural selection. So yeah, it doesn't make sense to say competition for unlimited resources. You have to competition for limited resources. So that's not the right answer. Gradual warming of the earth. I'm not sure what that has to do with natural selection here. asexual reproduction in species. That doesn't really make sense. Genetic variation. We talked about how genetic variation is kind of the basis for natural selection. So that's the right answer. Which of the following is not a requirement for Hardy-Weinberg? Remember, we talked about this. We don't want we want a large population because a small population will cause genetic drift. So that's the right answer. And then all of the other ones we talked about when I drew that diagram out, right? So small populations is the right answer. Okay. Common ancestor for both C and E, okay? So the best way to do this is kind of to draw the line and see where they meet. So C goes all the way down here, and E goes all the way down here. So where do they intersect? Well, they intersect at this node over here, which is number 4. So 4 is the right answer for their common ancestor. Yep, that's the right answer. Okay. Selective breeding of domesticated plants and animals to produce offspring with desired genetic traits. Selective breeding of domesticated plants and animals, okay? So this is artificial selection. It's done by a breeder, right? It's selective breeding. So it could also be stabilizing and disruptive and directional, but we don't know if they'd given more details on whether it affects, whether it reduces the mean or it extends, it increases the frequency of the mean, or whether it, shifts it directionally, then we can, choose one of these answers, but we don't know. So the best answer is the artificial selection because you have a selective breeder who's choosing, picking and choosing traits to select for. It's artificial. Okay. Hardy-Weinberg. This is in your formula chart. You should probably have your formula chart handy, the AP formula chart. So let's just go over this real quick. So Hardy-Weinberg's equation is essentially P plus Q equals 1. This is really all you have to remember. The second one you can easily derive from the first because for most of you who've taken algebra, P plus Q quantity squared is equal to P squared plus 2PQ plus Q squared. And obviously, since if I square the left-hand side, then the right-hand side is squared, and then that equals 1. That means this is the second equation. This is how I remember. So P is the allele frequency. Remember that. This is the allele frequency of the dominant allele frequency of the recessive allele, and then this is like if you guys, especially if you're in math and you've studied, binomial probability distribution, you know that this is like picking the dominant allele twice. And if you have the dominant allele twice, that's homozygous dominant. So this is the frequency of homozygous dominant. 2PQ is like saying, okay, well, that's once again, that's binomial probability. You could have dominant and then recessive, or you could pick the recessive first and then the dominant. That doesn't matter. Ultimately, that'll still produce a heterozygote because you have one dominant, one recessive. And then finally, Q squared is like saying pick a recessive twice, right? So picking a recessive twice is homozygous recessive. Q squared is the homozygous recessive frequency of the. This is a weird. I would specify, homozygous recessive genotype, but that's right. It would be this one right here. Yeah, be very careful whether it says Q or Q squared, because if it was Q, it's allele frequency. If it's Q squared, it's genotype frequency. Okay, I think this is a Hardy-Weinberg problem. "98 out of 200 experience the recessive phenotype." So Q squared is both the recessive genotype and the recessive phenotype, because the only way to get the recessive phenotype is through the recessive phenotype. Now, if it says that the dominant phenotype, then you have to be careful because the dominant phenotype is P squared plus 2PQ, because heterozygotes also exhibit the dominant phenotype. So if they give you the dominant phenotype, you want to subtract that number from 1 to get Q squared, the recessive phenotype. Generally, you always want to start with Q squared. So if Q squared they give us is 98 out of 200, what percent of the population would you predict would be heterozygotes? So we want to find 2PQ given Q squared. Okay. Let's just do some math to solve this. So first I want to simplify this down to like an easy decimal. So I'm going to divide both the numerator and denominator by 2. 2 times 4 is 8, 9 times 4 is 8, 2 times 9. Yeah, you can check that easily to calculate. 49 over 100. This is 0.49. So if q squared is 0.49, that means q has to be the square root of q squared, which is 0.7, I believe. So using that information, we know that p equals 0.3 because p plus q equals 1, according to Hardy-Weinberg. So 2pq is 2 times 0.7 times 0.3. This is 0.21 times 2 is 0.42. So it's 42% is the answer. That's correct. Okay. Which Hardy-Weinberg represents the genotype? Remember, p plus q equals 1 is the allele frequency. This we're asking for genotype frequency. So this is just if you just square the equation, then you'll get p squared plus 2pq plus q squared equals 1. That's it. So, yep. Okay. The frequency of heterozygotes. Remember, heterozygotes, you can either pick a dominant first, then a recessive allele from the gene pool, or you could pick a recessive allele and then the dominant allele from the gene pool. So that's 2pq, 2 ways of picking pq. This is literally a binomial distribution. So you can remember that way as well. But it's you don't have to go that complicated. That's just a more mathematical way of thinking about it. It's 2pq. Okay. Population of frogs has 37 individuals who are brown in color and 194 who express the dominant green color. Okay. 37 are brown, which is recessive because the green is the dominant color. So 37 is recessive. So 37 out of the total is q squared, right? Because that's the recessive phenotype or the recessive genotype. And then 194 is the dominant green. So 194 on whatever the total number is, that's actually going to be p squared plus 2pq because that's the dominant green color. So we want to find the dominant allele frequency, so we're trying to find p. Okay, so to find the total, we would add 37 and 194. If we add the two, I should be able to do this in my head. I'm not going to get out my calculator for this unless I absolutely need it. 7 plus 4 is 11. 3 plus 9 is 12. Plus 1 is 13. If you add 1, that's 231. So these two are 231. Just so I don't completely screw this problem up, I just want to confirm with my calculator. 37 plus 194 is 231. Okay. So q squared is that. I think the easier way, I wouldn't recommend using this. I would start with q squared, find q, and then subtract that from 1 to find p. So q is the square root of that business over here. And I would just do that in my calculator. Square root of 37 on 231 is 0.400 dot dot dot. So obviously p will be 1 minus q, which is approximately 0.6. I know I wrote equals. For the sake of this problem, it's fine, but you should probably write approximately. So 0.6 is right. Okay. Gene flow involves, we talked about this, gene flow has to do with migration. It's flowing from one population to another population. So it's individuals moving in and out of the population, migration essentially. So yeah, that's it. If a population has the following genotype frequencies, so let's go ahead and turn this into Hardy-Weinberg, variables. So AA is p squared equals 0.42. Big A little a is 2pq, that equals 0.46. And then q squared equals 0.12. Then what are the allele frequencies? Well, q we can find by taking the square root of 0.12. And that's approximately 0.35. The only answer choice that matches that is this one, but we can confirm p squared as well. We take the square root of p squared, we get p equals square root of 0.42, which happens to be approximately 0.65, so that's the right answer. In this question, we didn't even need 2pq simply because we were able to just use p squared and q squared. Now, say that they didn't give us q squared. You could have also figured it out by simply saying, okay, well, if this is 0.35, I can do 1 minus 0.35 to find out that p is equal to 0.65. So both methods work. There's a lot of ways to do Hardy-Weinberg problems, just like there's, it's a math problem. You can do it many ways. Usually some ways make more sense than others, and you'll learn that with practice. Genetic drift is likely to be seen in a population that's small. That's something that we just want to make that association immediately. Small is genetic drift, because you're more likely to have random changes in the alleles, allele frequencies, because you have a small population. Which of the following is an example of the founder effect? The founder effect has to do with genetic drift because you have a small population, because they just moved into a new environment, so you have a small population. But they're founding a new population, so this is probably the best answer. Yeah, because they're floating to an island and they're starting a new population. As the name suggests, they're a founder, right? So they're founding a new population. Yep. Which of the three graphs shows stabilizing selection? So remember, we talked about this. I think I might have said the opposite for what I meant. This here, I believe, is disruptional selection because it's disrupting the mean. This is disruptional. This is directional. Because this is directional because you have selective pressures pushing it this way, and then this is stabilizing because you have selective pressures on both sides. Actually, no, what I said earlier was right because it's stabilizing the mean. So now you can see that it's stabilizing the mean like that. So I actually think this is not the best representation. If I had to represent this, I would say if this is the main one, then the new one would probably look more like this. Reason being, if you've taken IB math, you know that the probabilities have to sum to 1, right? So that's an integral essentially in this case. It's normal distribution, and if the integrals are both 1, then the areas of them have to be both 1. And clearly in this diagram over here, the area under this curve, the dashed curve, is clearly less than the area under the solid curve. So that means that their areas aren't the same. So that means this is probably not an accurate representation. I would represent stabilizing selection like this if both of them are in the same diagram, just because we know that the areas have to both be the same because they're both 1. So if that, yeah, this is C, so C is the right answer for stabilizing selection. Let's check. And that's correct. Okay. Disruptive selection, we just talked about this. Disruptive would be A. Yep, and then directional would be B. Okay. If finches with large beaks and finches with small beaks increase in population, while finches with average-sized beaks decreases. So I like to think of this visually. So if I have a normal distribution, now large beaks and large and small increase, but not the average size decrease. So there's a selective pressure pushing down in the mean. So if it's pushing down in the mean, they'll start looking like this. And this is disrupting the mean, so it's disruptive selection. Yep, okay. Once again, the vocab isn't as important as the diagrams. Understanding the diagrams and the visual of what's actually going on is much more important. Okay, this is selection against the mean. We just talked about this. It's the exact same scenario as last time was disruptive. Okay. Robins typically lay 4 eggs. If more than 4 eggs are produced, then the baby chicks are malnourished, while smaller egg numbers result in no viable baby chicks. This is an example of, so if you have 1, 2, 3, 4, it seems that, so if more than 4 are produced, then they're malnourished. So that's not good. 4 is kind of your sweet spot, okay? If more than 4 smaller egg numbers result in no viable baby chicks. So there's a selective pressure for small, and there's a selective pressure for more than 4. So if I had to guess, I would probably say this is stabilizing because you're pushing against both extremes. So you're supporting the mean. I would say stabilizing, but we can check. Yeah, it is stabilizing. Okay. Human babies don't do well if they're born too small, and if they're too big, complications. So once again, Beyonce is having twins. I'm not sure why that's an answer. Okay, this is the same exact problem as last time, the last question. So this is also stabilizing. Yep. What type of selection is depicted? So this is directional because when you have selection against one extreme but not the other, that pushes the mean to the right or to the left, depending on which side it's pushed on. This is directional. Yeah. Father of evolution is Darwin. Mendel is the father of genetics. Malthus has to do with population. Watson, oh my, what did Watson do? I should know this. That's a little bit sad that I don't. Let me search him up real quick. You see the steam engine guy? James Watson? No, that's someone else. No, I'm sorry, that's completely wrong. Is it James Watson? Yeah, James Watson. Oh, of course, DNA. James Watson and Crick. That's right. Watson and Crick are with credited with the prize. They won the Nobel Prize, right? Yeah, Nobel Prize for their discoveries in the structure of DNA. So this would be Darwin because he's the father of evolution. He theorized natural selection. Whose theory included acquired traits such as a giraffe needing a long neck so that they stretch over time? I only know about Darwin. I don't know about Lamarck. So I would have just picked Darwin, but it's Lamarck here. So don't worry about that question. Lamarck must have studied acquired traits. Actually, let's search him up because it doesn't hurt to learn more. Obviously, you don't need to know this for the test, but he was a French naturalist, Jean-Baptiste Lamarck, and he was an early proponent of the idea that biological evolution occurred and proceeded in accordance with natural laws. Okay. That doesn't really help me figure out why this is the right answer, but that's the right answer for this one, acquired traits. Okay. Acquired traits are inherited would be Darwin because Darwin, oh, never mind. It's Lamarck here as well. Obviously, I haven't learned enough about Lamarck. We don't need to know about this. Don't worry about this. Yeah, these are all Lamarck. Okay, don't worry about that. Term given to describe the two-name system. We haven't learned about this, but it would be good to know it's binomial nomenclature because nomen means name in Latin. Bi means two, nomial means name. So it's a two-named naming system. That's what binomial nomenclature literally means. So it's that one. Yeah. What is the most important? Also, let me go back to that one. A good example of this, whenever you see something like H. sapiens or Homo sapiens, what that means is this is the genus and this is the species. This is the binomial nomenclature system. That's Homo sapiens is us humans. Okay. Most important phrase used in Darwin's book. We talked about this. It's descent with modification. So, the traits are they're passed down and then they, through variation, selection happens and modification. Descent with modification. Which type of speciation involves isolation due to behavior, such as sexual or habitat differentiation? Well, behavior or as sexual or habitat. So habitat differentiation is allopatric. The other stuff here with isolation due to behavior, that would be sympatric. So the answer choice here would be both. Wait, no, it's sympatric. Why is it sympatric? Let's see. Isolation due to behavior, such as sexual selection, or habitat differentiation. I'm not sure, because if you have habitat differentiation, you probably have a geographic barrier. Okay, I think that's not what it's saying. I think when it says habitat differentiation, I think it just means like kind of in the same vicinity. You have they're living in two different habitats. I'm not sure, but if there's a geographic barrier, that's allopatric. Otherwise, it's sympatric. Let me just confirm that with what we learned in class. The reproductive ancestral population. Okay. Oh, okay. This is actually important. I got this wrong. Okay, this is sympatric. Sympatric occurs from, so there's no geographic barrier. It can result from genetic mutations like polyploidy, or it can happen from habitat differentiation and sexual selection. So for sympatric, remember habitat differentiation and sexual selection. That's the right answer. Okay. What type of speciation involves geographic isolation? That's allopatric. We know that. Okay. The model that predicts evolution occurs at slow, steady rate, that's gradualism. We talked about that. Punctuated equilibrium is the contrast to that, which says basically we have rapid evolution that's punctuated, it's kind of interrupting, followed by long periods of stasis after it reaches like another equilibrium. So then more, then less. So sudden evolution, then no evolution, kind of for a long time. I think gradualism makes more sense. Obviously, punctuated equilibrium happens once in a while. I think gradualism is the bigger trend that happens. Okay, the model that predicts evolution occurs in rapid bursts followed by long periods of no change. We just said what it is. It's punctuated equilibrium. Okay, that's it for this Quizziz over here. Hopefully that helped. Obviously, you know, I'm the only one here right now, so I'll go through all of the questions, and that way, you know, if you have any questions, you can try and answer the questions for yourself, and then of course look at the explanation that I'm providing. Okay, so this one has 17 questions. Let's try this. So this is biotechnology mainly. Which method uses deoxynucleotides and northern blotting to determine the DNA sequence? Okay, well, we're probably going to have to use process of elimination because I don't know what the Sanger method is. This isn't recombinant DNA. Recombinant DNA is where you use the restriction enzymes, remember? There's actually a few ways of creating recombinant DNA. One is bacteria naturally do that. Let me look at my notes just to remind myself real quick. With bacteria, you have transduction, transduction where, okay, let's start with the other ones. You have transformation where you have the uptake of naked DNA. So transformation is when if you remember, so transformation is when you have uptake of naked DNA. What's that? So that's like when a bacteria just takes a naked DNA that's not protected by proteins or other molecules. Okay. Conjugation is the one I'm pretty familiar with. That's, remember, when you have two bacteria and they have these X pili, they can combine and then they can kind of, you know, share their DNA with each other. That increases variation. There's transposition, and then there's also there's transposition and there's also translocation. Sorry, not translocation, transduction. Transduction is transmission of foreign DNA into cell where viral genome. Okay, transduction has to do with viruses integrating their genome with the host genome. So okay, so let's just review that because I think I could use a review of that as well. So recombinant DNA is usually, that can happen. Okay, I think I got off topic. What we were looking at was the horizontal transfer of genetic information. Let's just look at that real quick. So horizontal transfer of genetic information happens in 4 ways. One is transformation. Transformation deals with what? Transformation deals with uptake of naked DNA. Okay, so it's not protected by a protein. That means transformation cannot be where the virus enters. This virus is protected by that RNA or DNA is protected by a protein. So it's naked DNA. The transformation could happen like if I were to just inject some DNA into a bacteria without any protein. That's transformation. Transduction is where a viral genome is like a virus. So you have a viral genome that's integrating with the host genome. Okay. Conjugation is where, you know, they're exchanging DNA, cell-to-cell transfer. So this is cell-to-cell. And then we have transposition. Transposition is the movement of DNA segments, transposition. That's, remember, like, I know in eukaryotes, we talk about transposition with how, you know, the bits of the chromosomes can be inverted, deleted, translocated, or duplicated. So those 4 ways of transposition in eukaryotic chromosomes. In bacteria, we have that, you know, 1 chromosome, and what can happen in transposition is, you know, their DNA segments might invert or, you know, translocate, that kind of thing. That's transposition. That's movement of DNA. These are your 4 ways of DNA segments. These are your 4 means of horizontal transfer. Now, what we're talking about, recombinant DNA, that's more dealing with the plasmids. That's what I was thinking of. Remember, we have the restriction enzymes that cut a gene, cut out a gene, and then it has sticky ends, right? So that those sticky ends now connect with the plasmid in the bacteria. So it might look something like this, and then this, so it might, yeah. So then if I have, so this might be the plasmid, okay? So let's say that this is the plasmid right here. how should I draw this? I forgot how I should draw this. So let's say that that's that there, and then so that's that. This is the plasmid right here. So you can see this part is a little bit overextended, and then this part is a little bit overextended. And then once the restriction enzyme cuts the gene I'm looking for, I can put that gene here with its sticky end. So the gene might look like this. And these areas here that are extra, these are the sticky ends. Those sticky ends are going to stick over here and over here. And now we've integrated into the plasmid. And now the plasmid, so the example of this is like insulin, right? So we can take out the insulin gene from eukaryotes, put it in the prokaryote, in the plasmid, and then using that recombinant DNA, we can have the prokaryotes produce the insulin for us. So that's recombinant DNA. PCR is polymerase chain reaction. That's where we use Taq polymerase, TAQ, Taq polymerase, to produce lots and lots of DNA from just one small. So if you look at a crime scene, if you have a little bit of DNA, I can amplify that DNA using Taq polymerase and running through that process over and over and over. And then that produces a lot of DNA from just a little bit of DNA. And finally, we have DNA fingerprinting. To my knowledge, DNA fingerprinting is we can do that through gel electrophoresis, where we load up DNA into the wells, and then here's what we have to remember with gel electrophoresis is that the negative terminal is close to the DNA initially, and then the positive terminal is on the other side. And the reason why is because DNA is negatively charged. So if we start this current, if we have a current flowing through these two electrodes, then what will happen? I'm going to show conventional electrical flow, not conventional current. Yeah, I'm going to show electron flow, not conventional current in this diagram. Then that causes the DNA to separate into fragments, and then we can compare the DNA segments with each other to see, if especially like in paternity cases, that's really useful. Let me just make sure that that's what DNA fingerprinting is. Let's see. Yeah, DNA fingerprinting is exactly that, with like the gel electrophoresis and having the DNA, separate into its separate fragments. The big thing to remember with that, once again, is that the negative is on the side of the DNA because we want it to separate, right? Because the negative repels the negative DNA. So, okay, so none of these are involved with these three that we know, so it must be the Sanger method. I don't know what the Sanger method is, but we don't need to know that. So let's skip that. Using living things or parts of living things to produce a product or solve a problem. Biotechnology, I think that's just a general definition. Yeah. Using a virus to enter a cell and replace defective genes with healthy genes. It's probably bioremediation. No, it's gene therapy. Okay, I didn't know that. So we haven't studied this, but it seems like gene therapy is where we do what? Okay, so gene therapy is where we inject foreign DNA or foreign RNA, foreign genetic material, and replace so that they can replace the defective genes. Okay. DNA analysis performed with forensic scientist. Okay, blah blah blah. 4 suspects. Okay, so this is your DNA fingerprinting, and this is pretty useful because now you can compare the DNA. Which suspect was most likely present at the crime scene? Well, clearly you can see that matching the crime scene DNA, the control is the suspect C DNA because those are near an exact match. So if they're a match, that means C was probably at the crime scene. So C. Okay. The first class of drugs developed to treat AIDS, such as AZT, were known as reverse transcriptase inhibitors. They work because so if they're inhibiting, they're stopping. Reverse transcriptase is what allows RNA. Remember, transcription is DNA to RNA. So reverse transcriptase is kind of the enzyme that allows viral RNA to turn back into DNA once it infects the host. So if it's inhibiting reverse transcriptase, it's inhibiting the virus's ability to turn from RNA back into DNA, so that infiltrate the host genetic information. So let's see. It's probably this one. Bonded to the viral reverse transcriptase enzyme, thus preventing the virus from making a DNA copy of its RNA genome. That would make sense based on what we just said, right? I think the other ones, Yeah, I think that this is the right answer because that's what reverse transcriptase does. Yeah. Which of the following is required to make complementary DNA, cDNA, from RNA? Well, we just said if you want to make DNA from RNA, you have to, go back in transcription, so it's reverse transcriptase. Which of the following is not true of restriction enzymes? Restriction enzymes are found in bacteria. Maybe that's true, I don't know. Restriction enzymes are needed for PCR. I don't think so, because if you're amplifying, so maybe this is not true. I don't know, we'll look at the other ones. Restriction enzymes cut DNA as specific regions, yeah, so remember the restriction enzymes are the ones that are kind of doing the cutting. Bacteria methylate regions of their own DNA that could be cut. I feel like that makes sense because, oh, that's an important concept because, oh, that's something we haven't talked about yet, and I'll introduce that right now. Methylation, this is important, is that methylation helps to make the DNA more tightly wound. Methylation makes DNA more tightly wound, and histone acetylation, so acetylation makes DNA, so this is for DNA, both of these. DNA, it makes it looser. So acetylation makes the histones kind of loose up, so the DNA becomes more loose, and then methylation helps the DNA more tightly wind around the histone protein. So, well, we know that this is definitely true, so we can cut this off. I think this could be true because I'm sure they could cut their bacteria or cut their DNA, so I'm sure they probably do have restriction enzymes. I don't know, are restriction enzymes needed for PCR? Maybe. I think they might be needed because if you have if I want to just copy this one gene, then I would probably have to cut it first and then replicate it. I would probably limit. I think it's this one. Reason being that why would you want to methylate your DNA if you're trying to cut it? If you're trying to cut it, you probably want it to be loose, right? So that you can easily cut it. If it's tightly wound, it's probably not going to cut in the right place because it's very concentrated. I would pick this one based on our definitions of methylation and acetylation. Let's check. Okay, so no, it's restriction enzymes are needed for PCR. So our initial instinct was right. Restriction enzymes aren't needed for PCR. I guess unless you have this long piece of DNA and you're only looking to cut that short piece. But really, I think PCR involves more of that Taq polymerase and that being able to replicate that DNA many, many, many times. Yeah, so bacteria methylate regions of their own DNA. Why would that be the case? Let's see. Oh, okay, that makes sense. That would be cut. So this is to protect against. So our reasoning was essentially right. It just wasn't applied properly. So bacteria protect their own DNA from restriction enzymes by methylating their DNA. So by making their DNA more tight, the restriction enzymes can't cut it properly. They're protecting their DNA. Okay, so good. And that would also justify this answer. That's why restriction enzymes are needed for PCR is the answer. It's not true for restriction enzymes. Okay. Which of the following explains how natural selection causes a trait to become predominant in a population? Okay, so natural selection, how does natural selection cause a trait to become predominant? So we talked about this already in the previous one, is how natural selection selects the more fit individuals who are able to reproduce and pass on those traits to their offspring. So individuals with traits that are favorable to the environment reproduce more, that's true. Organisms select which traits they wish to inherit. I don't think that's true. I wish we could all just, actually, I don't wish that. That's probably not the best thing to wish for. And then traits of an individual change in order to adapt. No. Adaptations aren't where individuals just choose, oh, it's cold over here, so I'm just going to grow some fur. That doesn't happen. It doesn't work. So the answer choice is this one. Individuals with traits that are favorable to the environment reproduce more. Okay. In a population of humans, the frequency of no dimples is 0.36. If no dimples is the recessive phenotype, so this is a Hardy-Weinberg problem. Okay. So no dimples is recessive, so phenotype. So recessive phenotype is Q squared. Q squared is 0.36. So they're asking the frequency of the recessive allele, which is Q. So Q squared equals 0.36, Q equals 0.6, because that's the square root of 0.36. That's right. Good. Okay. I think we already did this problem. If 98 out of 200, yeah, we already did this problem. I think the answer was 30%. No, it's 42%. Let's just do this. Let's just go ahead and work it out. Just because I don't remember it. So, like fully remember. So recessive phenotype is Q squared. Q squared is 98 out of 200. We're trying to find the heterozygous, which is 2PQ. So from this one, I think we said that this was 49 over 100, which is 0.49. Q is then 0.7, and because Q is 0.7, P has to be 0.3 because these two have to add up to 1. 2PQ is then 2 times 0.7 times 0.3, which is 0.42, which is 42%. Okay. It was the exact same question. I just don't have good memory regarding specific numbers. Okay. I think we talked about this already. A bird that can easily outcompete other birds for food and that can reproduce many eggs has a high fitness. So if they're more fit, they can reproduce more. Okay. What is suggested by the similarity of early embryos of different species of vertebrates? So remember, if you look at the embryos and they're very similar, that suggests that they probably have a common ancestor, right? Because they're similar. So probably recent common ancestry, because it could be evolution from a distant common ancestor, but I would probably go with this. Similar environments doesn't really make sense because embryos are something that's internal, so I think it would make sense to say recent common ancestry. Okay, never mind. It's evolution from a distant common ancestor. So we know common ancestry is true. I think the reason why this is the right answer is because it's asking about early embryos. If they were similar at late stage embryo, that would suggest that they're more closely related, right? Because they're not diverging as quickly. They're still the same for most of their time. I think this word early embryo suggests that it's distant common ancestor rather than recent common ancestor. Hopefully that makes sense. Okay, number 13. An organism has a vestigial structure. That structure once had a function in a probably an early ancestor, right? An early ancestor because just like your tailbone was a vestigial structure because our ancestors, our primate ancestors had tailbones, so that structure likely functioned in early ancestors. Yep. And the 5 factors of speciations are gene flow. Okay, so these are actually the 5 factors. I call these the 5 factors of the 5 mechanisms of evolution. We already talked about this with that table that I showed comparing the mechanism of evolution can be directly related to a condition for Hardy-Weinberg equilibrium. So, there's gene flow, genetic drift, mutation, natural selection, and the last one is sexual selection, which happens due to non-random mating. So if you have non-random mating, that means that, the boys prefer a certain variety of girls, the girls prefer a certain variety of boys. So that would be sexual selection. Okay. Mutations are important because they bring about genetic variation. That's a very fundamental idea. So it's genetic mutations have to do with genetic variation. That makes sense, right? Mutate means to vary, to change something. Which question are we on? Mutations. Okay, I think we're on 16. What is speciation? Speciation is when you have reproductive isolation, 2 populations aren't able to interbreed, and therefore they become separate. So that doesn't make sense. You can't combine 2 species to form one with different. That doesn't really make sense. Isolation that affects mating behaviors. It is isolation, but it's not necessarily that affects mating behaviors. Timing of mating that prevents. Okay, I think, this would be geographic isolation. This would be temporal isolation. And then this is speciation because it's divergence of 2 or more species from an existing one. So because an existing population, for whatever reason, it becomes reproductively isolated into 2 populations that are not able to breed anymore, then that leads to divergence. So it would be D. Yeah. One more question on this one. Which technology below would probably be the most important to a person who had diabetes and had to take insulin? We already talked about this, right? Recombinant DNA with the restriction enzymes, you cut off the insulin gene from the human cell, and then you use, you put that into the plasmid in bacteria, and you connect it using ligase, and then once that's connected, you have recombinant DNA, and now the prokaryotes, the bacteria, can produce the insulin for us, and we can just take that insulin. So that would be D, using recombinant DNA to produce human hormones from bacteria. Yep. Okay. Quite a bit of questions we've done already. Once again, there's 2 more here. I think there's no. We don't have to. I mean, yeah. Just because this is recorded and I'm not, holding anyone back, I'm just going to go through all of them so that if you'd like to go through them at a later time, you can. The more resources, the better. And the more practice, the better. Which of the following nucleic acids is not found in RNA? I know that this isn't on our test because we did this on our last test, but there will be questions on gene expression regulation, which will be on our test. So I still think it's good to practice these. Okay. Which of the following nucleic acids is not found in RNA? Thymine is not, right? Because RNA is AUGC. It's thymine. Which is the correct sequence of info flow? So this is the central dogma of biology. DNA to RNA to protein. Okay. Which of the following is not a component of DNA? Remember, DNA, nucleic acids have the sugar phosphate bone, right? You have the sugar phosphate bone. Sugar phosphate bone. I should actually draw it like this. If we were to think of it as kind of like a ladder, the rungs of the ladder are the nitrogenous bases. These are the bases, and then these are the sugar phosphate bones. So sugar is in there, phosphate is in there. Nucleotides are components of DNA. A nucleotide is when you have this much, when you have the phosphate, sugar, and then one base. That's a nucleotide. Yeah, uracil isn't found in DNA. Uracil is in RNA, right? Because DNA has ATGC instead of AUGC. Okay. Which aspect of DNA molecule allows it for it to replicate a duplicate copy of itself? Well, because it has a double helix structure, because it has a double helix, I can unwind that double helix and then I can replicate it. So that's why it's double helix. Okay, never mind. That's not the right answer. The answer is base pairing. That makes sense because the 2 strands are complementary because, complementary base pairing occurs. There's a complementary rule, so therefore I can, if I know what one base is, I automatically know what the other base has to be based on the base pairing. Okay, so I mean, I kind of, yeah, double helix structure kind of makes sense, but it's not the best answer here just because it's not the helix structure itself that lets it replicate. It's the fact that there's base pairing. That makes more sense. What is the name of the DNA strand that elongates away from the replication fork in a series of segments? So the segments are called Okazaki fragments, but this happens in the lagging strand, right? Because it's away from the replication fork. Remember, leading strand is towards the replication fork. This is lagging strand. This won't be on our test. We did this on our last test. DNA replication begins at short stretches of DNA with specific sequence of nucleotides called primers, right? These are the primers that start off. Maybe it's a template sequence that it's referring to. Yeah, I'm not sure exactly what this is referring to. It might be origins of replication. Origins of replication, like that replication forking, that's where you start. Or is it the template sequence? I mean, you have 2 template sequences. I think it's origins of replication. Yeah. Okay, that makes sense because the origins of replication have those specific sequences that allowed replication to begin. Okay. Which enzyme catalyzes the synthesis of new DNA by adding nucleotides to a pre-existing chain? Well, my tea is cold, obviously, because, well, I'm done. Anyways. The synthesis of new DNA by adding nucleotides to a pre-existing chain. Well, that's DNA polymerase. We should know that. That's the enzyme that does that. We can go through the other ones. DNA ligase helps connect those, those Okazaki fragments, those nucleotides. It's all about connecting. Helicase is what unwinds the double helix, and topoisomerase is that little thing that keeps the helix from winding back. And it's in front of the replication fork. It kind of helps keep it. It helps prevent the tangling. There's a term for that I forgot. Let me just search it up because that's going to bug me now. Topoisomerase. winding, okay, it's torsion, yeah. There's torsion ahead that would stop, yeah. So it prevents torsion, which is essentially the tangling that I was talking about. What property of DNA allows it to replicate, or supercoiling? I think that's what I was thinking about, supercoiling, yeah. What property of DNA allows it to replicate and repair? Base pairing does allow replication, specifically double-stranded structure. Well, you have to have double strand to be able to, unwind it, and then, replicate this one and replicate this one. And then, of course, it's anti-parallel, so that allows replication repair. So it's all of the above. Okay. I'm going to turn on my fan over here. It's getting warm. Okay. Which statement best describes the function promoter? So the promoter we just talked about in the very beginning with the prog was the promoter is that upstream sequence that that's where the RNA polymerase binds, right? Yeah. Yeah. And actually, now that I think about it, I think I said something wrong in the very beginning. I said that promoter is an upstream regulatory sequence. That's not true. Yeah, I remember what I said. I think I said that the promoter was an upstream regulatory sequence. That's completely wrong. Please forgive me for that. It is prog. Everything else is right. I'll fix that in the YouTube description. But you have your ROG and you have your gene. The promoter, that's where RNA polymerase binds. It's not an upstream regulatory sequence. You have other upstream regulatory sequences up here that are responsible for producing the repressor protein. Okay, I'm sorry about that. I will fix that in the description. I'll make sure to update that. Okay. During this stage of gene expression, genetic information is copied from the template strand of DNA. That's replication, right? Because if it's copied from the template strand, it's replication. What? Transcription, gene expression. I don't know. I feel like this is an ambiguous question. Copied from the template. Okay, I think the idea of a template strand is why we're calling it transcription instead of replication. Because if you have a template strand of DNA, that means that RNA in replication of DNA, you have two template strands, whereas if you have just one template strand that goes 3 to 3 prime. So the template strand is the 3 prime to 5 prime. That's the one that the RNA polymerase goes on, because RNA polymerase also happens 5 prime to 3 prime. Through the stage of gene expression, mRNA is processed through ribosomes and polypeptides are linked together into chains. So that's translation, right? Because that's when mRNA becomes protein, or yeah, polypeptide chains, and that's both. This process involves modifying the ends of the pre-mRNA molecules by modifying each end, cutting out introns. So that's, remember, that's splicing and then RNA processing, right? And the splicing that happens, remember the poly A tail modifies the 3' end, and then the 5' end has a 5' cap. And the reasons why we do that is so that, we don't want the hydrolytic enzymes to eat up those ends. So that's why we add those. So that's called RNA processing, all these things together. Okay. Which answer shows the correct sequence of information flow from gene to protein? So we talked about this. We have replication, then we have transcription. After transcription, we have RNA processing in eukaryotes, and then translation. If a DNA template strand has the following sequence, what is the correct mRNA sequence? So if I have AAA, remember mRNA will become U's, A's will pair with U. So we have U, G, C, and the only answer choice that matches that is this one. So I'm going to go with C. But just for practice, that's U, G, C, U, C, A. That would be it. Okay. Okay, so you want to remember the stop codons from the chart. You just want to memorize this UAA, UAG, UGA. We've already done this in the last test, but you still remember it. If the C codon has the following sequence, UCA, what's the anticodon? Well, the anticodon is just opposite, so it would be A, G, T, I believe. Yeah, AGT. What? Why is it AGU? Well, yeah. Oh, that's right. I forgot. Sorry, I'm so sorry. That's absolutely right because yeah, it's the anticodon also has to be made out of mRNA, so it can't have a T. Of course, it's AGU. Sorry about that. Yeah, it would be AGU. Okay. But DNA's template strand, if a DNA template strand's DNA sequence is AAA, what amino acid will be formed? The DNA is AAA. The RNA would be UUU. So then UUU, we look in the table. That's phenylalanine, I believe, PHE. So that's PHE. Mad cow disease causes normal versions of a protein in the brain to misfold, causing brain damage. So what's responsible for the change in shape of the protein? I'm not exactly sure. Is it prions? I think prions are the ones that are responsible for helping proteins. Yeah, prions are misfolded proteins. So that would be prion. Okay, prions are misfolded versions of protein which are infectious and cause brain disease. Okay, so that's just definition. Okay. Which portion of the brain is responsible for homeostasis? I don't think we need to know this, and I honestly don't know this. I think it's the cerebellum, but I don't know. Brainstem. Okay, yeah, I don't know this. We don't need to know this for the test. Yeah, we don't need to know anything about. I honestly don't know this. Cerebellum controls vision. I would think probably the. I have no idea. Frontal lobe. Yeah, no, occipital. Forget about it because yeah. Your grandfather started showing symptoms of muscle tremors, Parkinson's probably. Yeah, this is just a common genetic disorder. I don't think you would get questions like that on the AP exam. Individuals suspected having brain damage, verbal and written communication. We don't need to know brain anatomy, so I wouldn't worry about too much about this. Yeah, I really don't know any of these. I'm just taking guesses here. Okay, this is a good question. AP biology questions are generally like this. You get a little passage, then you have to answer the question. The fall of 1957, Hans Fromme was conducting research at a Frankfurt Zoological Institute in Germany. He noticed that several caged European robins were becoming restless and attempting to fly to the southwestern part of the cage. Fromme wondered how the birds knew which direction they were supposed to be migrating, since they were stuck in a darkened room and could not see any landmarks other than the stars. So if they can't see anything, but they're still migrating to the south, then that suggests that they probably have some sort of magnetic receptors that know the Earth's magnetic field. So that's probably an electromagnetic receptor. Yeah. Which type of signaling molecule is triggered by endocrine glands? Well, hormones, right? Oh, okay. This goes back to an earlier unit. So paracrine signaling is where you use local regulators and send those out. But endocrine signaling is long distance signaling, and endocrine system deals with hormones. Yeah, and yeah, they're secreted through extracellular fluids by their endocrine cells and the target cells through the bloodstream. Which of the following correctly describes? Okay, now I think we're getting into questions that have nothing to do with what's on the test, but I'm just going to do them for the sake of practice. Feel free to skip over these if you don't want to spend time. Endocrine system and nervous system, both are responsible. Let's see. Both systems send impulses directly. No, that's the nervous system. Both systems information can be received by various cells in a different way. I think the answer is D. Yeah, because they're both helping reach homeostasis. Yeah. Okay, which of the following gland is correctly matched to its function? I think this is the right answer because the pancreas releases insulin. The thyroid, not exactly sure. I just know the adrenal glands have to do with adrenaline, with like the fright response. Sorry, fight or flight response. Yeah. Oh, actually, that's wrong. What do the adrenal glands do? Let's see. So they help contain. Oh, I think cortisol is the stress hormone. So cortisol whatever gland releases cortisol would have been the correct answer choice for that. I'm not exactly sure about the functions of the thyroid. The thyroid helps regulate heart rate, blood pressure, body temp, and their rate. So that's involved in biological organs. Okay, well, nice to know. When a chromosome fragment reattaches to the original chromosome. Okay, this is good. We actually need to know this because we need to know about mutations. Chromosome structure can be altered by damages to the chromosome or during an error in meiosis. When a chromosomal fragment reattaches to the original chromosome in the reverse order, that is what? That would be an inversion, right? Because it's inverted. Translocation is where, you take one piece from one chromosome and put it on another chromosome, and they are exchanging segments. Deletion is, cutting it out, and the duplication is when you have a second copy of that. Mutation causes misplacement of structure, leg growing. Okay, mutation is likely to be what? So if it causes the misplacement of a structure in an organism, so if you have a leg growing on an antennae, I don't know what these things are, to be honest. P53 genes, I think, have to do with cancer, from what I remember. Oh, why are we? No, close that. That's not what I intended to do. Uh-oh. I think I shut out the whole thing. Let's just go back to that last page and just go back to that question. We were on something like 25. No, we were 7 maybe. Yeah. Okay, so P53 genes, I think, have to do with cancer. Yeah, tumor protein P53. Okay. RAS genes, I'm not sure. They seem familiar. I can't remember exactly what they do. Yeah, I think they're also dealing with cancer. Yeah, they're oncogenes. They have to do with cancer. Homeotic genes, I'm not sure what that is. I don't know what any of these are. Homeotic genes are what? Regulator genes so they have to do with body segments or structure. So this would be a homeotic gene then. Okay, let's also search up maternal effect genes because I'm just curious what that is. Maternal effect genes. So they're from the maternal genome. Products are required for the development of the embryo. Okay, makes sense. Okay, northern elephant seals. This is a good question for our, oh wait, we're not on that one yet. We're still on this one. Okay, 28. Mutation in the cell inhibits the production of tumor suppressor gene. So tumor suppressor helps prevent cancer. So if it's inhibiting tumor suppressant, then it will develop cancer. Yeah. What type of point mutation causes translocation to be terminated prematurely? So if I have a, so that means I have a stop codon earlier on, right? Stop codon earlier on will happen due to a missense mutation. Nonsense mutation is, actually, I'm not exactly sure. I can't remember exactly what those mutations are. So let's just search them up. Nonsense mutation happens, so it's a point mutation. Okay, never mind. So what we just did, so yeah, I said the wrong thing. Nonsense mutation is where it stopped earlier. So that's nonsense. Missense mutation is when it reads it incorrectly, right? This is just vocabulary. I just didn't remember this. Sorry about that. But missense is where, maybe you have a point mutation or something like that that causes, it to be misread or, yeah, nonsense means it doesn't make sense. So, okay, that's a good way to remember it. If it stops early, that's nonsense, right? It's baloney. That's right. Okay. Frame shift mutation, we talked about. Deletions, we talked about. Deletions, yeah. Okay. The type of mutation frequently alters the genetic message by changing the reading frame. We talked about that. That's frame shift because all the later mutations downstream are affected. Okay. What enzyme is responsible for the reverse information? Oh, we already did this one in the last one. It's reverse transcriptase. We can look at that problem in our earlier quizzes that we did. Okay, this is a good question. We should know this just from understanding Griffith's experiment, but we'll also take a look at the information here. What important conclusion did Griffith reach after conducting his experiment? I think it's this one. Living R bacteria. So the S is the smooth strain, R is the rough strain that doesn't have the capsule. The rough bacteria without the capsule had been changed into pathogenic S bacteria with the capsule by an unknown substance, and that's the transforming principle. So we can see that. You can read this on your own, but you should probably be familiar with the Griffith's experiment. This was on the last test, but once again, since we're starting to get close to the AP exam, it's good to start reviewing. Okay, which group demonstrates that DNA is the agent of information? Well, group 1 is just the control. It's not going to show anything. Group 2 is another control, so it's not the answer. Group 3, heat-killed S cells. That's not helping. Group 4 will demonstrate that DNA is the agent, because once you have the heat-killed S cells with the living R cells, obviously something transformed the R cells into pathogenic S cells. So that has to be group 4. Okay. Which type of bacteriophage reproduces only by the lytic cycle? I'm not sure. Lytic cycle is the fast, from what I remember, lytic cycle is fast killing and lysogenic cycle is slow killing, but I'm not exactly sure what type of bacteriophage would do that. Probably a virulent. Yeah, virulent just sounds like the right answer. Death of the host cell. Lytic cycle results from the death of the host cell. Yeah, I don't think we need to know this right now, at least, so don't worry about it. Bacteriophages are able to carry bacterial genes from one host cell to another, resulting in genetic recombination. And this process is called, so we talked about this earlier, right? There's 4 methods of horizontal transfer. The one where bacteriophages carry bacterial genes from one host cell to another that's conjugation, right? Because they're, exchanging genes through their sex pili. No, that's transduction. Sorry about that. I think we should probably review that, because I think I have a pretty bad understanding of that. Transduction occurs when bacteriophages carry Okay, sorry about that. Transduction occurs when bacteriophages carry bacterial genes from one host cell to another. This results from an accident during the phage reproduction. Yeah, I'm a little bit confused by this, to be honest. Let's just see, transduction versus transformation. Let's see what Khan Academy says over here. So conjugation, I kind of have a good idea, just because that's the one I'm most familiar with, where they're exchanging DNA through their pili, right? That one we know. But I don't know about the other ones. Okay. In transformation, a bacterium takes up a piece of DNA floating in its environment. Okay. Oh, okay, that makes sense. So you just have naked DNA floating around in the environment, and the bacteria takes it up. Okay. Transduction, DNA is accidentally moved from one bacterium to another by a virus. Okay, so a virus takes DNA from one bacteria and then puts it into another. Conjugation is what we said, and then we talked about transposable transposition. Transposition is important because that can provide antibiotic resistance, because they're transposition is the movement of the DNA segments, right? So yeah, transformation, that makes sense. So you just have DNA outside and that comes in. Transduction is where you have this viral DNA that's coming in, and now the viral DNA copies that, and now the virus might have actually taken some of the host DNA and then injected somewhere else. So it's essentially how DNA is transferred from one bacteria to another, but it's through a virus rather than directly through pili. Okay, so that makes a bit more sense. Why this would be because it's phages, right? It's the viruses. Bacteriophages are viruses. The viruses are carrying it from one host cell to another, and that's genetic recombination. So that's transduction. Okay, hopefully that clears up the confusion between those 3. Okay. All right, we've finished that one. Now let's go to Unit 7 with natural selection, and this is our last one for today. Quite a bit. I know we've been doing this for about 2 hours now, but the more questions, the better. You can always skip through, and yeah. Okay, so northern elephant seals were hunted by humans in the 1980s, reducing their population, but now it's over 30,000, but still they have reduced genetic variation, and that's because of what? Well, that's because of the bottleneck effect, right? Because because of human hunting, there was a bottleneck that kind of reduced it, and that resulted in genetic drift, which is low genetic variation. Yeah. Certain cave fish and salamanders have non-functional eyes, which are remnants of the functional. These leftover structures we know are vestigial structures, right? We talked about that. Which scientists tested the hypothesis that Earth. This is Miller-Urey experiment, where we talked about how you could have inorganic, you could have some, basic organic molecules like CH4, maybe maybe some methane, some phosphate, water, things like that. And then if you, strike it with maybe lightning or something with like electricity, that could produce, you could produce organic compounds from the inorganic compounds. You could produce like amino acids and stuff. So that's Miller-Urey experiment. Okay. What is the phenomenon that explains the sudden appearance of many phyla of living animals in the fossil record between them? So that's like in Earth's history. I don't really know, but it seems like it's an explosion of some sort of an explosion of population. So I would go with that. Yeah. Cambrian explosion. We haven't studied this yet. There's like a timeline. I don't think you have to memorize this though. I don't think that makes sense to memorize this, to be honest. Maybe we do. I don't know. First cells on Earth were likely to be prokaryotes. Anaerobic prokaryotes. We talked about that, and then they probably got engulfed by larger cells, and then that's how we got the eukaryotes through the endosymbiotic theory, which is not a step in the formation of the first living cells. Self-replicating molecules that made inheritance possible. That is true, because through the RNA world hypothesis, we suggest that RNA was a self-replicating molecule, and then eventually that DNA was more stable. So that is a step. So we wouldn't select that answer choice. Joining of small molecules into macromolecules, that is a step, because we want to start from small and build to get bigger. Biotic synthesis of small organic molecules, that was important. What about this? The packaging of molecules into droplets within membranes that maintain an internal, that is true as well. So one of these I picked wrong. So that's true. B is definitely true. So I'm going to eliminate B. The joining of biotic synthesis of small organic molecules. I think that's important, obviously. I think origin of self-replicating molecules is also important. I would select D, but I'm not entirely sure. I would select C, but I'm not entirely sure. So the correct answer is A. Biotic synthesis of small organic molecules. Why is that not a step? Let's see. Oh, because it's abiotic synthesis. I'm so sorry. Remember we just talked about Miller-Urey, right? Because how you can form, what Miller-Urey showed was that you can form organic molecules in abiotic conditions. So this would be abiotic synthesis. This is the phrasing here was wrong. Okay. Zebras' stripe pattern is determined by genes that turn on the production of melanin on only certain parts of the body. And then the timing differs between species, explains by different, okay. So for some reason, timing differs, and then different timings result in different phenotype. So I'm not sure what most of these terms are, but looking at the root words, chronological has to do with like has to do with time. So I would do heterochrony, because you have different you have different expressions for different chrono, so for different time. I would see. 3 million years ago, populations of snapping shrimp separated on either side. So this looks like geographic isolation. Okay, so this looks like geographic isolation. And that looks like allopatric speciation. Yeah, and because they're snapping at each other instead of the mating, they're not the same species, obviously. So it's likely allopatric speciation, because that's has to do, literally when there's a geographic barrier. Bowerbirds of Australia and New Guinea have one of the most unique courtship rituals. They make a bower with a colorful and unique object such as glass and pieces of litter. Okay, they're decorating themselves, so that's behavioral isolation. Okay, so evolutionary changes in mating rituals, like the differences, can contribute to which of the following? Well, it will contribute to speciation, probably. Well, no. So if they change mating rituals, I would say that if they're changing mating rituals, then that's probably speciation, because that's behavioral isolation, right? And behavioral isolation is a form of reproductive isolation, which leads to speciation, right? So I would say that that's B. Yeah, speciation. Okay. 3 spine sticklebacks were introduced into the lake in Switzerland. Since that time, they're splitting into 2 separate types. One is in the main lake, and now one lives in the streams. So this would be, I want to be careful here, because this isn't that there's this physical geographical barrier. This is habitat isolation, not. So they're both breeding in the same stream, same time of year, but they're splitting into 2 genetically and physically different types. I would say that this is sympatric, because this is habitat isolation rather than a physical geographic barrier. So this is sympatric, yeah. Western spotted skunks, not monks, breed in the fall, and whereas eastern spotted skunks breed in late winter. So that's temporal isolation, right? Yeah, because it has to do with time. Which of the following describes allopatric? So we want to see an example where there's a physical geographical isolation. I think it would be this one, but let me just read through the other ones. 2 different environmental conditions. Small segment of the original population. No. This would be sympatric. No physical barrier means sympatric. Okay, I'll go with this. Yeah. Okay. Wildflowers introduced, species crossed, they produce sterile hybrids. So when you have hybrid sterility, that's a post-zygotic, right? However, after about 2 new varieties, but they can't reproduce with any of the original 3, the mutation occurred. 2 or more sets of chromosomes, that will reveal the answer. When you have 2 or more, if you have additional sets of chromosomes, that's polyploidy, right? Polyploidy is also, that's another example of sympatric speciation, I believe. Let me just confirm that. Yeah, so if you have genetic mutations that result in speciation, polyploidy can be a mechanism of sympatric speciation. Oxygen revolution had an enormous impact on life. Remember, so originally we had anaerobic prokaryotes because Earth's atmosphere was mainly anaerobic. But then when you had oxygen, as oxygen started, being created, and the atmosphere became oxygen rich, that caused the extinction of many prokaryotic groups because they were anaerobic and they were not used to this oxygen. It is hypothesized that eukaryotic features evolved from prokaryotic cells when smaller prokaryotic cells, the precursors of mitochondria and plastids, began living. So we talked about this. This is endosymbiotic theory and endosymbiosis, right? There, endo means getting inside. Symbiosis means living together, not living together, but it's a symbiotic relationship. So where they're living together and it's a beneficial relationship for both of them. Endosymbiosis. Okay. In which nutrient cycle does bacteria play a key role in the conversion of essential organic compound from its atmospheric form to a form usable? So this we should just know. It's the nitrogen cycle. It's called nitrogen fixation. I think we've talked about this earlier, but bacteria in the soil help fix nitrogen so that it's usable by plants. Yeah. So remember, nitrogen gas can't be used by plants, so they help. It's helped, the bacteria help fix that atmospheric nitrogen into other forms. Male peacocks are famous for their colorful, showy tails. Female peahens are brown with no fancy tails. Okay. Sexual dimorphism. So when you have, so males are, so humans are sexually dimorphic, and many other organisms are. So which mode of selection produces species exhibiting sexual dimorphism? That would be sexual selection, right? Because if there's sexual selection, then there could also be, no, I think it's sexual selection. Yeah. Because it's selecting for particular sexes. Okay. The thermal regulation involves maintaining an internal body temperature in a tolerable range. Which of these organisms are considered ectothermic? I'm not really sure. Endothermic, I guess, would be internal heat. They're able to maintain internal heat. Ectothermic, I think, would have to be then like something like a reptile probably that's, you know, that can't maintain its internal heat. So I would think that that's probably the python. Yeah, they're getting their heat from the outside source, so they're ectotherms. What type of animals excrete nitrogenous waste in the form of ammonia? I'm not exactly sure. Mammals don't do that because actually they might, but to my knowledge, I don't excrete ammonia. I don't really know, actually. Let's just take a guess. I mean, I'm sure maybe you guys know, but I'd probably go with reptiles. That's fine. It's fish. Animals that excrete ammonia need access to lots of water because ammonia can only be tolerated. Oh, okay, that makes sense. So because ammonia needs to be diluted, we need the concentration to be low. So we want the volume of the water to be high. Which of the following does not describe natural selection? Okay. D is obviously right because natural selection can increase the proportion of favorable traits. I'm going to get rid of that. Individual organisms with certain characteristics are more fit for their environment. That's true. The organisms with the best suited characteristics will live to reproduce. That's true. So that is natural selection. Humans have modified many species by selective breeding. That's clearly the answer. Selective breeding isn't natural selection. It's artificial selection, not natural selection. Of course, higher fitness. As soon as you see higher fitness or favorable traits, that's natural selection. So the answer choice is B. Sickle cell disease is caused by having two alleles for hemoglobin S, and that results in blood cell that has sickle shape. So individuals who have one hemoglobin S allele and one hemoglobin B allele are said to be carriers. Although they have the sickle cell trait, they do not have sickle cell disease. Carriers are resistant to malaria. That's called heterozygote advantage because though the homozygous, so that's why this homozygous, the recessive allele stays, because even though the homozygous recessive is extremely fatal, that's what causes sickle cell anemia. If you have the heterozygote version where you have one dominant and one recessive, that can actually give you an advantage. It can help protect you from malaria. So that's heterozygote advantage. Okay. Okay. So this looks like it'll probably be, some sort of selection question. White-tailed deer vary in color from reddish-brown in the summer to grayish-brown in the winter. 1% of the deer exhibit a recessive genetic trait found in about 1% of all white-tailed deer called leucism. Leucistic white-tailed deer lack pigment over all or part of their bodies. They can have varying levels of white, from white splotches to being half brown and half white to near all white. Okay. So if I were to make this on a diagram, I would probably say that this is brown, this is half and half, and this is white. Okay. So this isn't like a numeric scale or anything. It's just arbitrarily I could have put white here and I could have brown here. It doesn't. Okay. This is, what I did here is just to help me visualize it. Okay. Leucistic deer is not very well camouflaged. So leucistic deer lack pigment over all. Okay, I think I'm doing this wrong. Let me read this question one more time. So they can vary from reddish brown to grayish brown. Okay. Leucistic traits, they're lacking the pigments. So leucistic deer lack the pigments. So they're not well camouflaged, so their chance of survival is very low. The color range of white-tailed deer, so these are the leucistic deer, can graph the following results. So this is, so it was right what I was doing before, because this is just leucistic deer. This is not the other deer, just leucistic. So since there's a stable mean here, this would be stabilizing selection because it's just half and half, right? The half and half is, white splotches being half brown and half white too. All white. So all white would be bad. White splotches would probably also be bad. The half brown and half white seems to be the best. I would go with stabilizing. Yeah. Because it prevents the intermediate phenotype, so it supports the middle phenotype. Just from the graph, you can tell that. Okay. Oh my. Okay. This organism causes causes a disease known as speckled leaf spot on wheat. This disease has the potential to cause significant grain loss if the environmental conditions are favorable to their spread. Scientists determined the genetic structure of this pathogen in various geographic regions worldwide and found that all populations collected from different geographic locations had similar frequencies of common alleles except the populations collected from. So these, I believe, are allele differences, not allele similarities, because these seem very low. Okay. The Mexican population here. Okay, so this is actually measuring genetic diversity, I believe. So it had lower gene diversity. Okay, we're looking at Mexico, lower gene diversity, few alleles at each locus, and the gene frequencies were significantly different from populations at other locations. The Mexican population was sampled for a breeding nursery using the screen for resistance pathogen. Nurseries located far away from the wheat population area and has limited potential for influx of natural inoculation. It was inoculated with a limited number of strains of the pathogen. Wow, this is a pretty hefty question. Okay, well, low genetic diversity suggests that low gene diversity suggests that it's probably some sort of genetic drift. So probably founder effect or bottleneck effect. But if it's, let's see, it's located far away from the wheat population area, it has limited potential for influx of natural inoculation, inoculated with limited number of strains from the pathogen. Few alleles. Okay. Seems, I don't know. This seems like there's some sort of bottleneck event here. Okay. Population has lower gene diversity. I'm not sure. Nursery is far away from the wheat population area. Okay, well, here's the thing. It's probably weird. It doesn't seem like they're founding a population. It's probably bottleneck because they were probably took out by much of the pathogen. I don't know. It is founder effect. Never mind. Let's see what their explanation is. Founder effect occurs when a few individuals have been isolated from a larger population and thus a smaller group of individuals establish a new population. Mexican population was isolated. Okay, so we knew it was genetic drift. I wasn't sure whether the nursery had, like, whether it was actually, okay, I guess it implies that it was a founder effect because this nursery is located far away from the wheat population area and has limited potential influx of natural. Because it's isolated and it's far away, whenever you have isolation far away, that suggests that it's a founder effect. Yeah, in this case, especially when there's small population and that's genetic drift, right? Okay, in Australia, only a relatively small number of individuals arrived on this continent with the introduction of modern agriculture, and therefore, so small number of individuals means, and they're arriving, so that's founder effect, genetic drift, low gene diversity. So I would probably choose C, low gene diversity consistent with founder effect. Yep. Geographic area with the highest level of gene diversity is likely to be the center of the origin based on the data. So we want to see the highest one, the one with the highest amount. That seems like it's Israel. Yeah, Israel seems to be the highest level of gene diversity. So I would go with Middle East. Yeah. Study published by the team led by USGS revealed the movement of polar bears towards areas with more persistent sea rise. Okay, so sea is rising, polar bears are moving towards, more persistent sea ice. Completely wrong. Where there's more ice, polar bears move towards that because they don't want, more water. They'd rather live on land. Researchers defined 4 general polar bear population clusters. Okay, looking at the genes, they found that the polar bears have recently begun to move in a northward direction from South Canada. Okay, so they're moving further north. That makes sense, probably because the sea levels are rising, and therefore they want to move further north where it's colder, right? The team of researchers hypothesized that the Canadian archipelago will be a future refuge for polar bears if climate change continues toward a warmer climate. The movement of polar bears population provides a prime example of I don't know if it's founder effect here because I don't know if they're moving into new. No, they're going. So the ones that are below are going into the Canadian archipelago, which already has people or already has a polar bear population there. That's migration. It's not necessarily founding a new community. But because it's migration, that's gene flow. Yeah. Which of the following was not a large-scale process which influenced major evolutionary changes in life on Earth? Okay, continental drift and mass extinctions were definitely large-scale processes. Okay, adaptive radiation is probably also a large-scale process because obviously. So we didn't talk about adaptive radiation yet, but that's we talked about it in class. But that's where organisms like species will adapt to fit different ecological niches. They will adapt to fit to fill in those roles in the ecosystem so that you have a stable ecosystem. So that does seem like a large-scale event. The founder effect doesn't seem like a large-scale process because that happens with small populations, right? And that's genetic drift. Yeah. Evidence from which of the following does not support evolution? Okay, homologous traits do support evolution because homologous traits deal with a common ancestor diverging based on different environments. Of course, natural selection supports evolution. Fossil record supports. Analogous structures are what don't support evolution because analogous structures just show that there's convergent evolution because two species or populations from different ancestry come together because they live in they need similar functions because of the environment that they live in. So it's analogy that doesn't support evolution. Fossil record is a significant record of the history of life, but it's an incomplete record. Okay. Many fossils are destroyed, okay, and many are yet to be discovered, so it's biased in favor of species with certain characteristics. Which of the following is not a characteristic that would be included in the fossil record? Species that were abundant. Well, they would probably be included because if they're abundant, you're probably going to find their fossils. If they had hard shells, probably because they're not going to be destroyed. Species that existed for short periods of time. That's probably not something that would be included just because they're not long. They're long enough to be deposited and found. Species that were widespread, obviously that goes with A. So the answer is C. In animal development, which stage of embryonic development involves the rearrangement of the blastula into a 3-layered embryo? I'm not exactly sure. Fertilization happened earlier. Organogenesis, 3-layered embryo. So organogenesis sounds like the organs are being developed. I'm not sure what gastrulation is. Let's search up what gastrulation is. Cleavage sounds like something's being cut. It's cleaved, so that doesn't sound right. Gastrulation is the blastula reorganized into multi-layered cell structure. Okay, it is gastrulation. Yeah. Organogenesis is when the organs start to form. Which group of tissues or organs most likely derived from the ectoderm? Okay, so these are stuff we probably don't have to know for the test nor for the exam, just because this, or maybe we need to know it for the exam. We just haven't learned about it. I don't know. Derived from the ectoderm layer during embryonic development. Ectoderm probably sounds like epithelial lining of the mouth and anus because those are outside. Yeah. Unique family of genes in animals that plays in development of embryos. Controls the expression of, I think it's Fox. Nope. Okay, we don't know about that. Don't worry about that. Which group of organisms share these 4 key characteristics at some point? I do not know. Dorsal, post and post-anal tail. This sounds like vertebrates. I don't know. I really don't know. Nerve cord. Okay, if there's a nerve cord, that's probably vertebrates. Never mind. Chordates. Chordates is the phylum to which vertebrates belong. Okay. We probably don't need to know this in much detail, as much detail as this, because I don't think we need to memorize every single phylum. Which of the following is a longitudinal flexible rod located between the digestive tract and the nerve cord? Once again, I don't know. I'm just gonna go with I don't know. Doesn't matter. I don't think we need to know this. Which groups of mammals lay eggs? Those are monotremes. That's just something I knew. Platypus and spiny anteaters. Those are your monotremes. Generally, mammals don't lay eggs, but they're characterized as mammals for other reasons. Okay. Mule deer live and feed in predominantly open areas in the mountains of Western North America. Okay. Slightly less food available in the forest edge. However, the risk of predation is greater in the. Okay, the reason why they don't live on the forest edge, even though there's more food there, is because they have more predation. Their predators are more over there. So this is kind of a cost-benefit compromise, and that's favoring behavior that minimizes the cost and maximizes the benefits. Sounds like an optimal foraging model. That seems like the best answer here. I don't know what the other ones are. Yeah. Because it's optimizing foraging. That makes, I think, the wording gives it away. Some animal societies, such as honeybees, a single female mates with several males. That's poly. It's not. It's not. How do you pronounce it? Monogamy. It's not that. It's not polygenetic. That doesn't make sense. What's the difference between polygyny and polyandry? Let's see. It's one of those two, I think, the answer. But let's look at the difference. Polyandry. Polyandry is woman has more than one husband, pattern of mating in which female has more than one male mate. Polygyny is where a man has more than one wife. Oh, so it's like Ginny, like we know the term misogyny, something like that. So Ginny has to do with woman. So polygyny means man has more than one wife. Polyandry means female has many men. That's actually the root word for that. I think that would actually help me remember what polyandry is. I'm sure that andry part has something to do with, yeah, ander in Greek, aner or andra, it means male. Okay, cool. As tadpoles change into frogs, their tails get smaller, cells die. That's apoptosis, programmed cell death. We learned that much earlier in the year. Okay. Although the leaves of a pitcher plant, Venus flytrap, and a cactus do not appear to be very similar, they are derived from a structure present in a common ancestor. So if they do have a common ancestor, that means that that's homologous, right? Because it's diverging evolution, not analogous. Analogous would be similar function. Same structure means common ancestor means homologous structure. Yeah. Which description best describes a cell-mediated response to pathogen? A cell-mediated response. So the cell is responding to a pathogen. So defenses such as skin, that doesn't seem like a cell-mediated response. That seems like just external kind of barriers. The proliferation of B cells. And to be honest, I don't know in detail B cells and T cells, but I think that would be the answer. I think B cell. What do B cells do? B cells make up antibodies. B lymphocytes are part of the immune system. Okay. B cells make antibodies. I think that would make sense. Antibodies defending against infections and body fluids. I would go with, okay, with cytotoxic T cells. We haven't learned about that. We'll learn about it later. Cytotoxic T cells attack foreign cells. So yeah, we'll learn about those later probably. You don't need to know them for the test on Monday. Okay. Saliva, tears, epithelial tissues, and the mucous membranes make up which part of the mammalian immune system? They're barrier defenses because they're, outside. Body cell does not have the protein called a class 1 MHC molecule. It's likely that it's been infected by a virus or converted to cancerous state. Which type of cell patrol is the body? Attaches to cells without class 1 MHC molecules and kills the cell. Probably killer cells. Yeah. I don't think memory cells or histamines make sense. Interferons, I'm not sure what those are, but natural killer cells makes the most sense. Which type of response to a pathogen in plants leads to cell and tissue death near the infection site, thereby preventing the spread of the disease? I don't really know. Probably systemic acquired. Yeah, no, it's hypersensitive response. Plant. We don't need to know this yet, by the way. Let's not worry about this one yet. Which example illustrates two? Okay, most recent common ancestor. We should show which of these has the most recent common ancestor. Okay, I'm going to immediately eliminate D because just because they have fin-like structures to help them navigate through, That could be convergent evolution. That could be analogous structures rather than common ancestry. New World vultures and Old World vultures look very similar. Both have featherless necks and heads, feed on carrion. They only use visual senses. Okay, but I think that this could also be wrong because this might be convergent evolution simply because that might be convergent evolution because they have similar niches. Wings of birds and insects perform the same function. No, that also seems to be convergent evolution. Hawaiian silver sword plants are very dramatically in their distance, in their appearance. Some are tall, twiggly trees, blah, 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 but their genes are very similar. As soon as you see genes are similar, that's common ancestry, and that provides evolution. That provides evidence for evolution. That's A. Yeah. Okay, look at the 4 phylogenetic trees. Different evolutionary history for taxa A through D than the other 3 trees. For taxa A, D. Okay. Here, I'm not exactly sure what this means. Well, in either case, A seems to be the output. I think that that's evident. B and C seem to be, okay, so in these, okay, I think the answer to this would be A. Reason being, if you look at these 3, C and D are the most similar, C and D are the most similar, C and D are the most similar. And in either case, A relationship to D is the same in each case, and they're just, flipped around the nodes. So here, however, B and C are the most similar. So I would go with A because that's the one that's different, 6 up. Based on this phylogenetic tree, which statement is not true? Okay. What does this show? Well, crocodilians are a sister group to the group containing pterosaurs, ornithorian, dinosaurs. I'm not sure what sister group is, but that's probably not true. I think birds and Saurischian dinosaurs are what probably would be characterized as a sister group because they're like, next to each other. Orthisian dinosaurs and Saurischian dinosaurs are more closely related to birds than lizards and snakes. I feel like that would make sense. Okay, so since it is true, it's asking not true, I'm going to cancel this out, eliminate it because, yeah. The lineage leading to the lizards and snakes was the first to diverge from the other lineages. That would make sense because it's the first to diverge from the other ones. Crocodilians are more closely related to ornithisian dinosaurs than to lizards. That probably doesn't make sense. This is probably the one that's not true. How is this true? Let's see. Crocodilians are a sister group to the group containing, okay, that makes, so it's crocodilians are the sister group, the sister group to the group containing the pterosaurs. That would make sense. Yeah, so I think the answer choice, so we could eliminate this one because that is true. The answer choice would be D, that's correct. Okay, we talked about this. It's do kangaroos prefer cake or frosting, generally speaking? The answer choice would be, but it's most inclusive to least inclusive. So most inclusive would be the biggest, so it would be A. You just have to remember that. We're using an acronym. Mine is, do kangaroos prefer cake or frosting, generally speaking? I think there's another one. Did King Philip come over for a glass of, I don't know, something. You can make something up. Which domain has the following characteristics? Single-celled, no internal membranes. Okay, single-celled is probably prokaryote. We do know some single-celled eukaryotes, but if there's no internal membranes, that's probably prokaryote. Now, here's the thing. Prokarya isn't a domain. Eukarya is a domain, but the 2 prokaryotic domains are bacteria and archaea. Okay. Now, if they contain peptidoglycan and in their cell walls, that's probably bacteria. Yeah. Archaea do contain introns and some genes, and they don't have peptidoglycan, like we said. We'll learn about this next unit probably. So I don't think you need to know this for the test, but it's, once again, good practice. Which kingdom has the following characteristics? Heterotrophs or autotrophs. Okay. Pseudopods, cilia, or flagella. So these are probably small ones. So, plants don't have cilia and flagella. If you see pseudopods, that's probably protists. And they're Vitus variety of eukaryotic. So yeah, that's probably protists. So that's, yeah. We'll learn about that next year. Okay, I think we're done. All right. That's quite a bit of stuff we covered today. Quite a bit of questions. We did a lot today. You know, feel free to just skip through and see what you need to work on, and you know, you can leave a comment or just text us on our Slack and ask, you know, if you have questions. Please subscribe to our YouTube channel and follow our social media for Revise Now if you like. And good luck on the test.